All right, I suppose it's time that we get started. So hello, everyone, and thank you for joining us for day 2 of a 2-day conference on the present state of biomarkers of human longevity, hosted by the international nonprofit consortium and decentralized longevity industry knowledge and collaboration platform, longevity.international. My name is Franco Cortez, Director of Aging Analytics Agency, and I will be your moderator for today's talks and discussions. Biomarkers of human longevity has been a strong strategic priority for Aging Analytics Agency and its parent consortium, Deep Knowledge Group, for a number of years now. Besides being one of the global longevity industry's most underrepresented yet disruptive sectors, we also see it as the lowest hanging fruit and the most ideal solution for a number of important areas of the industry's continued growth and stability, and as the critical catalyst for developing a more sophisticated method of validating longevity therapeutics and lifestyle interventions, for optimizing investment decision-making, translating longevity R&D and science into practice, for formulating and executing relevant, socially impactful longevity policy and governance, and as really the only tool that is market ready today for measuring, mediating, and adjusting factors impacting personal, practical human longevity. And it is against this backdrop that we will be hosting today's talks and discussions. So day 2, sector spotlight and diversity of practical applications, will expand upon topics discussed during day 1 yesterday, and will focus on developments in specific subsectors and use cases of the biomarkers of human longevity industry in a deeper manner. Today, we'll be hearing from a number of industry thought leaders and executives working at the forefront of biomarkers of human longevity, each of whom will be presenting their latest findings and achievements, followed by a panel discussion led by our very own 𝐁𝐻 to explore the topics of today's talks in a deeper and more dynamic manner. To start us off for today, Ryan Smith, founder at True Diagnostic, will be presenting his company's Dunedin Pace Epigenetic Aging Clock. We'll then be joined by Professor Eveline Bischoff, a longevity physician at Human Longevity, who will be presenting on the clinical application of human biomarkers of aging and their role in longevity medicine, followed by Dr. Peter Fedichev, co-founder and CEO of 𝐁𝐢𝐸, who will be speaking about the quantification of aging from big longitudinal biomed data. Following Peter, we'll be hearing from Dr. Afshin Beheshti, a NASA Ames Research Center scientist, who will be talking about mitochondrial dysfunction as being a central driver of health risks associated with spaceflight, and I assume some interconnections relating to the aging process. Following Afshin, we'll hear from Philip Metras, co-founder and CEO of Evocal Health, on unlocking the human voice as a biomarker for health and longevity, followed by Anastasia Georgievska, CEO and co-founder of 𝐁𝐢𝐸𝐴I, on the role of gender-specific scales in performance evaluation. Following Anastasia, we will hear from Kashif Khan, CEO of the DNA Company, on hacking your DNA to reverse chronic disease and slowing aging, followed by Jingjong Shi, founder and CEO of Ginfinity Precision Medicine, on personalized optimization of NAD and health via laboratory testing. Following that, we'll hear from Jonathan Stein, CSO and Medical Director of Testis Laboratories, on NGS and telomere testing in anti-aging diagnostics, followed again by our very own 𝐁𝐻, who will be presenting an overview of longevity femtech biomarkers. And after Kate, we will be bringing everybody together to launch a panel discussion that again will explore the topics of today's talks in a deeper and more dynamic manner. And that panel discussion will be moderated by 𝐁𝐻. And a reminder for everybody tuning in today who may have missed day 1 yesterday, everybody who's with us today will be sent video recordings of that, as well as today as a follow-up. And I'd like to specifically highlight the fact that we released 2 rather important publications yesterday, which I encourage you to check out. The first is Aging Analytics Agency's newest open access special analytical case study, Longevity Biomarkers Landscape Overview Q4 2021, produced for Longevity International, and that is available in an open access format for all of you to review at www.longevity.international/biomarkers. During yesterday's proceedings, we also launched the newest book by Deep Knowledge Group General Partner and Aging Analytics Agency founder, Dmitry Kaminskiy, titled Biomarkers of Human Longevity, Data Science for Accelerating Aging Research and R&D, and the Critical Catalyst for Practical Human Longevity, tangible investment decision-making and de-risking, which again was officially launched during yesterday's proceedings and is available as of this hour on Amazon and at longevity-book.com/biomarkers. And I've also been informed that this book will also be distributed to all of today's participants, attendees, and registrants as an interactive full-color PDF, free of charge, and a follow-up email. So look out for that. Without further ado, I would like to welcome our first speaker for today, Ryan Smith, founder at True Diagnostics, who again will be presenting on the topic of his company's Dunedin Pace Epigenetic Aging Clock. Ryan, it's a pleasure to have you with us. The floor is yours.
Yeah, thank you all so much for having me. I'm excited to talk today in a great line of speakers. What I really want to talk about today is just a little bit of an introduction to sort of who we are at True Diagnostic. And then in particular, I think one of the best aging algorithms that exist out there in terms of reading DNA methylation for aging-related appearance, and that is that Dunedin Pace of Aging metric. And so just to give you a little bit of background on us, we're a relatively new company. We started offering commercial methylation epigenetic testing and reporting in July of 2020. However, since then, we've developed one of the largest private epigenetic databases in the world with over 15,000 patients with multiple very exciting covariates, like full-scale proteomics, full-scale metabolomics, and a wide variety of phenotypes and other lab testing. We're really excited to use that type of data, that large-scale data, to then create new and better ways to interpret the epigenome of every individual, and not just for necessarily longevity-related concerns, but things even beyond that, by looking at things like Alzheimer's, the imprintome, newborn screening, and et cetera. Currently, though, our focus is definitely aging. We have over 30 clinically approved trials at the moment where we're investigating the effects of several types of interventions and looking to create new helpful aging-related algorithms, such as senescent burden level detectors, among others. And we do a lot of that work with researchers as well. So if anyone ever has any interest in doing any specific methylation research, I would highly encourage anyone to reach out to us as we're definitely investing into this field. In addition to that, our commercial offering is an at-home blood test where we will take sort of a fingerstick lancet. We'll run an epigenetic analysis of over 900,000 CPGs and then report that out mainly in age-related reports. And so our age-related reports encompass a little bit of everything. They encompass things like telomere length, immune cell subsets. So you can look at things like the percentages of each immune cells, like your CD4 and CD8 T cell ratio. We do the overall biological aging, separate that between intrinsic and extrinsic aging. And we do this pace of aging, which will be the main point of topic today. As I mentioned, the way that we're able to create some of these new and innovative algorithms is by a lot of that covariate data we're able to collect from our physician group. It's really, I would say, our one sort of secret way to really push the field of aging diagnostics further by gathering a lot of covariate information from all of the physicians who are using our testing. And we are particularly, I would say, interested in looking at these multi-omic related values. So looking from everything from genomics and epigenetics to transcriptomics to proteomics, metabolomics, and ultimately those phenotypes we're trying to predict. And so that is the approach that we're taking, and hope to have several multi-omic oriented aging clocks out in the middle of next year. And so really the algorithm I want to talk about today, and one I think is most exciting for our purposes, is this Dunedin Pace of Aging metric. And so unlike traditional biological clocks, this is not reading out an overall age. What it's reading out is essentially your instantaneous rate of aging. So at the time of testing, how fast are you aging? And this has several benefits we'll discuss today as we go along, but we'll be answering sort of all of these different questions. And so before I go into the actual algorithm, one of the things I really want to talk about is how these algorithms are created or trained. Really in the first generation clocks, which were made popular by Dr. Steve Horvath in his 2013 work, what they were sort of training against or predicting against was chronologic age. So they were taking all these large scale methylation data sets in order to predict the chronological age. And that was a really important step, and one that I think is fundamental for the aging research we're doing now. However, these algorithms over time changed a little bit to these second generation algorithms. And these were not trained to predict chronological age. What these were trained to do were to predict markers of morbidity and mortality, so clinical biomarkers. And this is a lot more helpful in the medical space, because instead of just trying to predict a chronological age, if you usually wanted that from a patient, you could typically ask. However, what we're really trying to do is project how rates of aging might affect risk of disease. And these became a lot better with these second generation clocks. However, now this sort of after sort of advancing from these biological clocks like PhenoAge and GrimAge, the health and retirement study done at USC sort of listed this Dunedin pace of aging as a third generation clock. And the reason being is this has been trained on the longitudinal cohort and is able to capture a more instantaneous picture of aging. And so in order to give a little bit of background on this, it's important to know some of the players involved. This was mainly developed by Duke University, and it really started with a cohort from the University of Otago in New Zealand. Really, when Duke first applied for funding for this, they were hoping to look at a cohort and measure their aging throughout an entire lifespan. And actually, when they first asked the National Institute of Aging for financing on this, they were sort of laughed at. They thought that the National Institute of Aging thought that there would be no variation in aging rates in younger individuals, and if they did, it would not be significant to the phenotypes that they would develop later on. New Zealanders who were born in 1972 and were 3 years of age in 1975 when the cohort started. And in this cohort, they measured a lot of really interesting age-related phenotypic biomarkers. They measured everything from postural hypertension to DEXA imaging scans. They did auditory testing. They did markers and measurements of physical function, such as 30-second chair stands. They did even optic nerve scans, as well as dental imaging and even advanced MRIs. And so some of the data that was used in this cohort is really advanced data sets. And again, this is on a longitudinal patient population. So they started with when they were 3, and now these patients are over age 45. And so with all of that data, they sort of defined this 4-step approach. The first was to collect this longitudinal data, then to second, create a pace of aging based on those clinical biomarkers. And with that pace of aging, they then were able to create a methylation-related diagnostic via elastic net regression modeling, which is the method used for most of these methylation-related algorithms. And so you can see here, these are the 19 different blood-based biomarkers that were used to develop this phenotypic aging score. And this is sort of how the mathematical process they use to create that score. And so as you can see here on this x-axis in the graph, to the left, you have little to no biological aging. And as you get further on that x-axis, you would have faster chronological aging. And some of the patients they looked at in this cohort were aging up to 2 years per chronological year. So over 20 years, they might have aged 40 years biologically in this cohort. And so being able to track those differences in aging were fundamental to develop this pathway, which ultimately led to a DNA methylation algorithm which could predict the aging rate in individuals and predict phenotypes outcomes as well. So once they developed this, the idea was how would they validate the Dunedin pace algorithm in different cohorts? And so they've looked at several different studies to do this. However, I really want to talk about some of their results at age 45 here in to sort of a way to illustrate how clinically relevant this algorithm already is today. So some of the things I like to show here are some of their performances at age 45, sort of segmented by their rates of aging. One of the things I think is constantly a battle that we have to fight in this longevity space is sort of this false dichotomy between health span and quality of life and overall longevity. I think a lot of people equate older lifespans to poorer quality of life, but these things are not mutually exclusive. And I think that this is some of the phenotypic testing which proves that. If you look out here on this x-axis, you can see the rate of aging. And as you can see, the performance on balance testing was significantly worse with the faster aging. And this type of arrangement did not just happen with balance or physical performance testing. It also happened with things like grip strength or muscle mass and body mass sort of metrics like frailty. It even extended to looking at cognitive decline, where as people who were aging at a rate of 2 biological years per year were on average around 5 IQ points lower than those who were not. In addition to that, we saw a lot of changes in MRIs of the brain as well, where we saw cortical thickness diminished and surface area diminished in older individuals as well. And then lastly, we can even see this on facial aging appearance. And this is one of my favorite things to sort of show, because a lot of times when we talk about aging, we talk about relative risk, and relative risk can be a little bit hard to motivate some patients. And so a lot of times we like to show this picture as well. So these are facial generated images of everyone in the cohort at age 45. On the left, you have the 10 slowest aging members of the cohort as detected by this Dunedin PACE algorithm. And on the right, you have the 10 fastest aging members of the cohort. And so what you can see here is that all these people are the exact same age. The difference between the youngest and oldest members of the cohort looks to be a 20-year difference, at least visually. And the way that this is true is that, in fact, they probably have aged that much more due to their faster rates of biological aging. They might have been aging at a rate of 2 biological years per year, while the other people in the group, the slowest aging members, might have been averaging right around a 0.6 biological age per year appearance. And so what this goes to show us is that the faster that we're aging, the worse our performance on all of these phenotypic measures, and including facial aging, meaning that we're aging from the inside out, and that age is a predominant factor driving all of the development of these diseases. And so this is a really, really great clock because it's able to predict and pick up a lot of those phenotypic comparisons. But in addition to that, it has some other benefits compared to some of the other clocks. So one of the things we like to point out in comparison to the other clocks, and this is a published paper from that same group at Duke, shows that there's not a whole lot of overlap between some of the other age algorithms, particularly the chronological age or biological trained algorithms, which suggests that this is measuring a different process of aging than what might be captured via some of those clocks. In addition to that, this clock is really exciting because it has a relatively low standard deviation, meaning that we can get very, very precise measurements. This is definitely even highlighted a little bit further by looking at something called the interclass correlation, or the ICC values. And really, to define an ICC value, it is essentially how correlated are one result to the other. So if we took the exact same sample and we measured it twice, how correlated would those results be? And obviously, you want a result that's as close to 1. You want it to be almost identical to if you're taking the same sample. And so here you can look at some of the most popular aging algorithms and their relative ICC values. You can see with the 2013 Horvath clock, for instance, it is reading right around a 0.675 ICC value. And the beauty of this particular Dunedin PACE algorithm is that it is substantially higher, and the only one of these tests which has a high level of precision with an ICC value over 0.9. This is changing, and thankfully, one of the biggest problems with these methylation-related age diagnostics is their variability and some of the noise you might see in those algorithms. And so the Dunedin PACE definitely has the best ICC value of any published algorithm to date. However, some of the work that Morgan Levine is doing at Yale with Albert Higgins-Chin is also very exciting, as they're using principal component analysis in order to make the variability of all of these aging metrics significantly lower. But right now, this Dunedin PACE represents one of the most precise measurements, which makes it great for personalized medicine. What we can see in some of our patients and our physicians who are using this testing is that they might have the same treatment protocol for 2 different patients, but each of those patients might have vastly different responses to their rates of aging through this measurement. And so you can get a really good idea of what is working for you to get this as low as possible. One of the other really exciting things about this is that we're also seeing that it works and performs very, very well in saliva as well as blood, which will be able to sort of roll out a saliva-based method of pace of aging detection, which will be relatively low cost and highly precise. And so if we look at the benefits of this algorithm, by far the #1 benefit is the precision. Compared to a lot of these other aging algorithms, we can see even statistically significant interventional results in a period of 3 months, where that was very, very difficult and required very large sample size with some of the other age algorithms. In addition to that, it's detecting instantaneous rates of aging. So if, for instance, if someone has lived a very poor quality of life from a health perspective previously, and then they try and change their lifestyle to have optimal aging, we can actually see that change happen rather than still detecting some of their other previous behaviors. And again, this is an algorithm that's been trained longitudinally off the same patients, and so it's not confounded by generational exposures like the prevalence of antibiotics or even leaded gasoline, for instance. And so this represents several features of these aging clocks where there's some significant and vast improvements. And in addition to that, we know that this is incredibly predictive. We already talked about all of those phenotypes which it influences in a positive a negative way. If you can get this below 1 and keep this below 1, it is very, very good for your overall health. Even if you're slightly above 1, even if you're aging at a 1.01 biological per year basis, you would actually increase your risk of death over the next 7 years by 56%, and you increase your risk of a chronic disease diagnosis by 54%. So these risks are very, very predictive and give you a great target to try and keep this below 1 as much as possible. So with that being said, that's a little bit about the algorithm, but I also want to talk about a little about what is the newest version. This Dunedin PACE only came out in the beginning of 2021, although we're definitely already using it in several investigations to find out a little bit more. And so the first thing that I should say that it has taught us is that the impact of early life is incredibly important. Sort of now, this sort of innovative thought process in aging is that early life adversity can affect this rate of aging, which then proceeds and might predispose people to worse aging-related phenotypes over time. For instance, even measurements of brain function at age 3 in this cohort were predictive of aging-related outcomes at age 45, meaning that aging rates can be set in early life and in utero. And this is backed up by several other studies as well. Particularly, we have looked at this study, which shows that the Dunedin PACE is accelerated in early individuals who have experienced early life adversity. So for instance, socioeconomic class is highly correlated with more affluent populations scoring better and having slower aging rates in this metric. And we can actually even detect polyvictimization in this as well, which would then negatively impact that rate of aging. And in addition to that, we see this correlated and corroborated with another study, which looks at the Dunedin PACE as it relates to neighborhood level or family level socioeconomic class, where lower socioeconomic groups have worse rates of aging, which sort of lead into this idea that our aging rates are set even at very young ages, and that aging should not be something to consider at 40 or 50 years of age, but throughout the entire life process. In addition to that, this is able to pick up, I would say, socioeconomic disadvantage in a way that no other clock is as well, which provides some additional benefit there too. And I know we're running a little bit tight on time, so I'm going to go through these next slides a little bit quickly. But we also know that there are some validated interventions to change this metric. That's sort of the number one question we get asked is, what do we know about what has been published to change this metric? One of the things that we know based on this calorie study is that caloric restriction, particularly 25% caloric restriction over 2 years, can vastly change these rates of aging. As you can compare the non-caloric restricted group on the left to that graph, and then the caloric restricted group on the right, and their relative change in biological age over the course of 2 years. In addition to that, we also know that the Dunedin PACE, or as you can see it here, the Dunedin POEM4X, is by far the most sensitive at actually looking at the effects of these interventions. So it was actually able to capture the anti-aging effect of caloric restriction better than any other age algorithm. And I think this is true for most scientific research investigations. In addition to being a great tool for personalized medicine, it's also a great tool to incorporate into research trials to try and look definitively at what is having some of the best rate and change in some of these measurements. And so, we've also then applied this to things like cognitive dysfunction, where this is best at predicting and picking up changes in cognitive dysfunction or mild cognitive impairment or dementia. And it performs a little bit slightly better than GrimAge on that as well, which has typically been the best. And we've also applied this to things like COVID-19 and mRNA-based vaccines, where we've seen some very interesting results, which I probably don't have time to talk about today. But for instance, we have seen that people over 50 with COVID infection tend to age worse, and people under 50 probably have a hormetic response due to their naive T cells, which actually confers an anti-aging response. And we also see significantly shorter telomeres as measured through our telomere-linked estimators through methylation as well. We've even applied this data to dasatinib and quercetin-related senolytic applications, and had great results at protocol over the course of 6 months, reversals in every age-related metric. And we're applying this even beyond this to several other clinical trials, which you can see listed here, and really hope to report on how this metric is changing the rates of aging, and really what we can look at from population level scales to make recommendations to really affect the phenotypes of aging. And so I know that was a lot of information, but would love to open it up for any other questions I might be able to answer.
Thanks so much, Ryan. Very interesting content indeed. For anybody who has follow-up questions for Ryan, I'd ask you to pop those into the Q&A box at the bottom of the screen, and we'll do our best to address them during the panel discussion later today. It is now my distinct pleasure to welcome to the floor Professor Eveline Bischoff, longevity physician at Human Longevity, who will be speaking on clinical applications of aging and their role in longevity medicine. Eveline, it's a great pleasure to have you with us today. The floor is yours.
So once again, thank you very much for having me. And it's a great pleasure to talk today about the clinical application of the human biomarkers of aging and their role in longevity medicine. That was a title that I chose, probably a little bit unfortunate because being in such a panel with so much of vigor and enthusiasm, which is absolutely correct and which we are living also every day, my topic will probably bring a little bit more of a duster part, meaning what is happening at the end chain of human biomarkers of aging and their actual application in a daily life on patients. And as a disclaimer on myself, I'm an internal medicine doctor besides of being a longevity concierge doctor. So I am with one of my feet also still in the academic setting of an university hospital, currently here in China, pretty much still in the setting of internal medicine and also intensive care. So seeing longevity medicine and the rapid development of this fantastic field, and I hope soon to also be a new discipline of medicine, I also see what is going on in the traditional part of medicine, in the reactive part of medicine. And I am always wondering, how can we bring both together? How can we implement more of the longevity medicine, especially the measurements of the biomarkers of aging in the clinic, in the actual current clinic, because this will, at the end of the day, perpetuate the field of longevity medicine and ultimately bring benefit to a number of stakeholders, not only individuals, but also population, nations, and so on. So what is longevity medicine? I think there is no definition that is written in stone. We've tried to at least give a little bit of a guidance in our recent paper with Professor Xiaovoronkov and Dr. Kai-Fu Lee, the father of AI, basically defining the longevity medicine as a branch of precision medicine that is specifically focused on promoting the healthy lifespan, so the health span and the lifespan powered by AI technology. And we are looking at in longevity medicine, as opposed to traditional medicine, is the following. So in our traditional view of medicine, not in traditional Chinese medicine, but in a traditional view of medicine, we are following the patient's journey along the lifespan. And we are usually seeing that patients at different types of ages, so being in the children part, in adolescent, then at the peak of their performance, they are being accompanied by physicians that have, of course, a limited scope of expertise, but what is even more important, they have right now tools that are allowing them to look only at a very specific range of people, of populations that are falling into the same age range. So bottom line, we in the clinic, in a normal clinic, we are looking at ranges for a specific group of chronological age, because this is what we have been taught. This is what we are basing our evidence on, the randomized controlled trials. So this is the old school. And what is the problem here? Even if we would change the chronological scale axis to biological axis, this would still leave us at looking at the doctor at the range of the patient's age compared to the population, and dealing with the patient, usually helping as a reactive medicine when the diagnosis of some sort of disease is there, or even at the early prevention, but still looking only at the frame of that person. In longevity medicine, the goal is a little bit different. Here we are looking at the same patient journey. So of course, one of the goals of longevity medicine is to hopefully extend the healthy lifespan, but it's not the primary goal to only extend the lifespan. We already have this with our medicine. The lifespan of humans has been extended and brought with it the problems that we are dealing with, namely the age-related diseases and frailty and so on. So what longevity medicine is doing is to look at the person's range of the optimal performance. So identifying the biological age of a person where he or she can perform best. And the performance is not only the subjective measurement, so it's not only when the patient felt best and so on, but really the best performance also speaking biologically and physiologically, etc. And being now able to process the data with AI, with deep learning, transfer learning, and so on, we can identify at a specific point of time the biological age of a patient, and not only that. So knowing the biological age or knowing something is one thing, and it's very important. And thanks to that, we can now conduct studies that will teach us more and that will ultimately lead to identification of new biomarkers and application of therapeutics. But many patients in longevity practice are asking, all right, so now we have identified the biological age of mine, what next? And the answer is coming exactly from this type of learning, from deep learning, and from the outputs that are being provided. Namely, we can now receive from the algorithm an objective measurement of the parameters that have to be optimized, and an exact number of how much those parameters have to be optimized to bring the patient from their current state of biological age to their biological age of optimal performance based on the current biological age. So very often a person that is 70 will not have a calculated optimal biological age of 30, but maybe it will be somewhere around 60 or 55. And this is the goal of longevity medicine, to realistically and objectively, with quantifiable biomarkers, bring the patient to the state where he or she can be of optimal performance. In terms of aging, this usually means with no comorbidities and so on. So this brought, of course, the discipline and the field of longevity to be a new reality. And this also brought a new specialty, hopefully soon to be a specialty, of a longevity physician to the state. And it is important to remember that longevity physicians also have their place in the clinic. They are all under Hippocratic oath, and they also have the same goal. They want the optimal health for the patient. However, the way how the data are being curated and analyzed and finally also used and applied in following and leading the patient is very different. So this is why it is so important also now that physicians from different disciplines, since longevity medicine is so extremely inclusive, and I think it's the most inclusive discipline with dermatology and endocrinology and all the other types of disciplines together. It is extremely important that there are sources for the physicians to learn and to become well-trained longevity physicians. Now, longevity medicine is based on two pillars, in my opinion. That's just only how I see it. One is a clinical precision diagnostics, and this is where a lot of things is already happening, and we have all these wonderful tools out there, from monitoring and blood tests and all the other testings of multimodal diagnostics that is out there, more or less accessible, more or less available, but it's out there and it's progressing very rapidly. And then there are longevity interventions, and this is something that is fueled usually by doctors, and of course accelerated by stakeholders and by the patients and their motivation and so on. But this is the arena that is a little bit slower and also a little bit immature. Now, myself, another disclaimer, I'm in China, in Shanghai, and what I have seen here is something that we could see worldwide. AI is definitely transforming the entire world. And we have seen being here, the entire evolution of how AI was changing the world up until the point of 2015 around the deep learning revolution that allowed us longevity physicians now to have the tools that we have. But what is more important, and I'm saying this also in relation to biomarkers, at the end I will say why it's important. So here in China, we have also seen how AI permeates the society and how in waves it came to be that right now we are pretty much living in a whole AI world, up to the point that the autonomous AI took over a lot of branches of life. In China, this includes, of course, medicine, up to the point that we have now over 300 purely internet hospitals, and almost all hospitals are going digital. Why is that also important? Because this is also transforming the human intelligence. So also this is transforming the maturity of the population per se, then the patients, on how they perceive longevity, how they perceive AI, and how they perceive AI also in their medical treatment, assessment, screening, and so on. And last but not least, we see that here the number of science is absolutely booming, the number of publications, etc. And this has been ongoing since the early 2010 and so on. So around 2014 already here, we could feel as doctors very much the shift from reactive to proactive medicine, with a lot of telemedicine implementation and a lot of curating of data. Still quite chaotic and discoordinated, but there was already this trend that was ongoing. Then we had the deep learning that came on top, and we were able then to really use some sort of preliminary inputs of objective function and lead to some targeted optimizations of our clinical questions. So what we see now in the past years, and the COVID very much accelerated this, is really the movement not only from reactive to proactive, but from proactive to AI precision medicine, to not only prevent some of the diseases, but really to mitigate, to identify the risk, to mitigate the risks of ever developing the diseases. And this is the trend, and I believe this is something that we will see also globally, or are seeing globally. So I believe most of this development will be at different pace, also in different ways of how it will be done, but also on other continents. Why is it important? Number one, because all of the physicians have pretty much the same goal. They want to optimize patients' health, and this is where a lot also of efforts of other stakeholders are in. But this is also especially very much important because we are looking at the silver tsunami. As we all know, we are having the fastest growing segment of population being the old patients, and they are extremely complex, not only for medicine, but also for society, for politics, and so on. So in terms of biomarkers of aging, there might be, or we might be facing very soon, also an increasing push from the public health and global health and governance sector to support really the actual translation of all those biomarkers that are out there and the measurements that are available into the clinic. And maybe we'll be soon looking at even screening programs for healthy longevity biomarkers, because we will not only want to prevent specific diseases like cancer, colon cancer, breast cancer, and so on, but we will probably also want to prevent aging or accelerated aging. And another point is more on an individual level. So people are now more aware that there is a high level of biovariability, and that this biovariability can be measured, and there is an increasing individual demand for curating the data, for sourcing of solutions, and for an actual translation for application of those. And of course, the increasing longevity interest is also perpetuating a field of point of care centers and point of care, up to the point that we have also at home solutions for a lot of data that can be curated by the patient, him or herself. So this is a very positive development. However, it needs the more a coordinated and credible effort of translation. And this can and should really happen in the clinical setting. So as to summarize, the need for application is definitely there because there is a global public health motivation, there is a personal individual maturity that we have arrived to, and we need to curate better data sets to create better algorithms that we have now, and also to have a clinical validation, not a spotty one with, but also a coordinated randomized controlled trial ones. Why? Because this is still fortunately or unfortunately how the clinics at the end of the day are running, and the medical field at the end of the day is able to move. Without RCTs, without credible clinical validations, there cannot be a good progress, and everything will be left in more of a dispersed setting. And this will also perpetuate the research and ameliorate the sampling methods that are also still quite spotty, and the availability and access to the test. So also the accessibility, and here also a point of equality. And last but not least, we need coordinated efforts. As one of the example that I would like to mention is oncology, or the field that is newly developed that I am very passionate about, the geroncology, so the oncology of the old cancer patients, where we have almost zero guidelines, but at the same time, we know that this is exactly the part of the population that is at the most risk to develop cancer, because age or old age is the #1 risk factor to develop cancer. And the incidence is now already around 60% of those who are over 65 years of age, and this will only be growing. And another reason for why oncology is so interesting and important in this setting of biomarkers is because there is just so many pathways that are parallel or interconnected between senescence and oncology. And last but not least, it is extremely important because oncology is associated with therapies that are usually causing something that is related to aging. Namely, very often we don't know that the patient is frail or at the edge of frailty. And then one destabilizing event, such as chemotherapy or radiotherapy, can bring the patient from a state of apparently healthy and young to frail, although chronologically young. So there is a big need of good biomarkers in oncology or in geroncology, and we have seen already in studies previously that that patients who have undergone oncological treatment are actually at a higher biological age after that, because they have damages that that were at the level of the hallmarks of aging that are accelerating it. And there are some very preliminary advancements in that field to find biomarkers. For example, P16 made some of highlights, but we are still in children's shoes, as we would say. And we know that the amount of biomarkers is, of course, growing. So there were efforts in the teams already in 2012, 2015 to somehow categorize them and bring them to some clinical translation. These efforts were still very preliminary, but quite good in my opinion, because they were trying to disperse the biomarkers into disease fields so that the clinicians could use this classification and apply it based on what we do in the clinic. And what we do in the clinic is basically looking at the ICD code and at the patient, and then by the coding, we can actually move and act. But there are challenges, and the challenges are definitely some of the typical ones. So lack of validation in very old people, because those are not included in the trials. There is a poor comparability and then poor way of pulling the data. And last but not least, it is very challenging. And there was also even a question by the authors: is it really useful to have some sort of a healthy aging score? Would it be useful? Well, several years after, we saw that yes, it would be, and today we have many more biomarkers of aging. This is pretty much the standard that doctors are looking up for. It's the list of AFAR's biomarkers of aging today, both listed on the website. But as we can see here, those are not exactly directly translatable to the clinic. And then we look at some of our other colleagues' work, for example, for Alexey Moskalev, where we see a huge abundance of biomarkers today. When we zoom it in, we see that here we have biomarkers that are still, measurable to some extent by the great progress of startups and companies that are making them available, but they are by far not the day-to-day life in regular clinics. And what is very important is also to remember that biomarkers of aging also include other measurements besides of blood. So we have an abundance of signs, vital signs, and other measurements that are important. And those are being measured in a much higher frequency. Optimally, as a longevity physician, I want to see my patient having a 24 hours monitoring of blood pressure, pulse, and so on. So how do we implement that as a biomarker into our algorithm and keep it together in the clinic? Also markers such as peak expiratory flow that are quite subjective. So how do we actually find solutions that will give us at least some sort of sensitive outcome that will be then longitudinally trackable? So surely AI will help us in that, and in the field of longevity medicine, AI-powered tools will help us to generate objective functions and actual answers to questions. And also in longevity medicine, we are looking at the aging at different levels. So this is giving us some sort also of order on where we put our biomarkers to, and then select those that we can apply in the clinic. So what is happening now, for example, in the centers that I work in human longevity, we are collecting around 150 gigabytes of data of every single assumably healthy patient who just came for the clinic. And this includes also precision imaging. This includes also multi-targeted body functionality, and of course, the entire multi-omics. And all those biomarkers are being collected and being stored. Some of them immediately have some output and actionable items are being developed for the patient, but some of them are not. But the real value in collecting all those data is, of course, as we know, the further learning. What is happening immediately is something that, for example, I do with my patients, is developing a longevity protocol that is applicable for that patient and is very individualized. And where are the biomarkers fitting in here? This is, for example, a granular schedule where different frequencies for different modalities of testing are being prepared for the patient. And this is, of course, evaluated and re-evaluated very frequently to obtain longitudinal data that are needed, and also for events that are needed at a specific time. And we are more and more also looking at some new biomarkers of brain age and cognition. And this is how we are proceeding then with diagnostics and monitoring to reversely collect more and more data to fuel our algorithms and give even better predictions. And just as a last example, I personally also, and there is no interest of commercial whatsoever, but I like to use deep aging clocks for the short-term monitoring of a patient, because this is giving me quite an immediate output of what I can do and what I have done for the patient in terms of getting them biologically younger and reverse their biological age. So this is an example of a patient who was presumably healthy and started own regime of NAD boosters and metformin and rapamycin. Typical patient who is young, in his 40s, very athletic, but unfortunately without a guidance. And what he has done actually in his own manipulation was to make him biologically older. Why? Because we know now, and this is what we also learn reversely using those deep aging clocks, which parameters are being overexpressed or being damaged in the body by an overuse on some of the current available geroprotectors. The same we see also for example, these young females that are biologically older and have reproductive challenges, where we then have to study and look further on which of the parameters besides of the blood and their interrelations are actually working. And this is how it all comes together. So the patient is collecting data, him or herself at home. There is a lot of markers that are being updated and checked regularly by myself or other colleagues. And then, of course, there is a protocol of medication and protocol of collecting new biomarkers and screening to, at the end of the day, quantifiably see if we are successful or not. And at this moment, of course, we are using different clocks, but here it's very nicely seen that this patient, for example, has aged chronologically over 3 years, but biologically we have kept him at the same level. So it will not be only a subjective feedback of the patient that he feels better, looks better, and so on, which is very flattering and great, but this is a quantitative, objective measurement of a real success or failure of the longevity protocol that the patient is on. All right, I hope I didn't go too much over time. Thank you very much, and happy to take any questions after.
Our pleasure, Dr. Bischoff. Yes, I found your presentation both deeply insightful and broadly applicable, which is a fairly hard dual optima to juggle and to maximize. And I must say a very high degree of alignment between our group's thoughts on challenges and opportunities and your own, especially as it pertains to the need for multimodal biomarker data and the fundamentally necessary as opposed to complementary role that AI has to play. So thank you very much. And anybody who has questions, please pop those into the Q&A box at the bottom, and we'd be happy to cover them during the panel discussion. I would now like to welcome to the floor our next speaker, Dr. Peter Fedyshev, co-founder and CEO of 𝐁𝐢𝐸, who will be speaking on identifying the minimum set of biomarkers of aging in humans. Peter, welcome. It's a pleasure to have you. The floor is yours.
Well, thank you very much. That's the first time I'm talking at your event, and I'm happy to do that. So I represent 𝐁𝐢𝐸. 𝐁𝐢𝐸 is a biotech that is aiming to defeat aging. And the specific way we're doing that is by studying big biomedical data from humans. We use AI and good old school physics in order to understand the character of human aging from lots of data, and then overlay that with molecular and genetic information in order to understand the key regulators of human aging. So I think you have seen that all. We know that not only our chances to die, but also to acquire major age-related diseases increase exponentially as we age. That deeply suggests that there is a common underlying process. And of course, we're interested to learn if we can measure something on human beings right now, or maybe in subsequent steps in not too long of a time in between the measurements, in order to see what kind of an aging trajectory is being developed or how to intercept it. Why is this important? Is that because we have an inspirational example, I like to show it all the time, that there are animals that don't seem to age. There are mammals like naked mole rats, and this is the work by Kalinskiy. These people have measured mortality of these animals as function of age, and on the bottom right chart, we can see that the chances to die in these animals do not increase as they age. So the challenge, I think, to the field is not only to recognize if certain things, guys, girls, animals, age or not, or do not age at a certain rate to measure the pace of aging. The fundamental question is that can we distinguish these two situations where aging occurs, let's say, normally, or can we build biomarkers that can also feel if aging in these animals do not actually happen or happen in some other way? So these are basically the questions which we are trying to address in our work. We work with leading laboratories. We work with Brian Kennedy, Andrei Gudkov, and other people. We have lots of access to human data from UK Biobank. We have access to animal data. And in what follows, I will try to give you a certain, let's say, the summary of observations over the last few years with surprising result. So first, what is the biomarker of aging? We have seen lots of examples already today. There are multiple hallmarks of aging. There is an increasing field of study. There are more and more hallmarks of aging. which means that most probably those hallmarks of aging as well as chronic diseases have common mechanisms. So in fact, this is known in other fields of studies, most prominently in engineering studies. When complex systems decide to disintegrate when a catastrophe is developed, most of the time you observe two common features. So first, prior to the catastrophe, the system develops slow motion. So the system becomes the system becomes slow. It's been frozen. And second, that slow motion acquires very high amplitude. So you see at the same time, first the system freezes, and at the same time you see the increase in amplitude of the fluctuations. So that's why we look at data from aging animals or humans, and we use machine learning, we use deep learning in order to find features that exhibit exactly this behavior. So we use longitudinal measurements, which means that we have the same guy measured few times along the course of their lifetime. And we try to compress all biology, transcriptomics, all kinds of omics, all kinds of signals. Since the hallmarks of aging are correlated in between each other, we try to compress all this information into as few numbers as possible, for example, 1, and use that number, that only number, in order to predict the future state of that animal. In such a way, you don't need to know if this animal is old or young. You don't need to know if this animal is going to die or not. You don't actually care. The only thing what you are trying to achieve here is to build a network, which is a future telling machine. This network is trying to take the information from now and use it to predict the future state of this animal. So just for fun, I will show you what happens if you do such a program to mice. So you use, for example, blood from mice. You build that single number, which is just a network property. And then on the left chart, you can see that this number is a function of age. So the number is DFI, the age is the horizontal axis. You can see that this number is increasing with age, as it should be. It increases with age exponentially, as it should be, because we know that animals and humans age exponentially. And the doubling time, the rate of this exponential growth is actually the same as the mortality rate doubling time. The most interesting set of points on this graph are the red dots. So the red dots are the biological age. Let's call this thing the network property of the biological age. This biological age is from groups of animals that are scheduled to be killed in the laboratory because they are too frail. So essentially, you can see that it doesn't matter how old or young you are. If you reach that ultimate biological age value, you're going to be killed because of the ethical considerations. So the mice in the laboratories are dead by achieving a certain cellular level in the biological age. So far, so good. I'm reminding you that we only use the trajectories. So we are using AI in order to learn individual trajectories of aging animals. And we have already found something that describes aging in these animals, which is totally good. So as I promised to you, since all the hallmarks of aging are correlated, all age-related quantities in mice should correlate to that network property, to the biological age. And here we're measuring the number of senescent cells in these animals. So these are luciferase reporting mice. So they're glowing. You can actually measure how old they are by observing the number of senescent cells physically. And you can see that there is a very good correlation in very old animals, the correlation between the quantity that we're calculating from blood and the total number of senescent cells in these animals. So the biomarker like this kind of biological age works very well. You can see, for example, that if you put animals on a high-fat diet, the male mice actually suffer. They live shorter, but also their biomarker levels is also smaller, lower at already very young age. So in short, you can use machine learning and common sense arguments from physics and engineering sciences to use longitudinal data to actually learn, literally learn aging trajectories, if you like. In such a way, you get a number, let's call it biological age, that describes aging totally cool. It has all the relevant features. It correlates to all hallmarks of aging. So in a way, the network gives you the mother of all hallmarks of aging. And at the same time, you can use it in actual aging studies. And you can start getting surprising results. For example, high-fat diet kills males, doesn't kill females. I don't know if it actually translates to humans, but that's what we can do with biomarkers if they are set up well. You can use it to test your drugs. For example, this is a typical experiment that we are doing. We are using this network output in order to see if a drug is working or not. You can see here an example of an intervention that is applied for 6 weeks only. You can see that in the blue, in the treated group versus the orange control, you can definitely see that during the treatment, the biological age is going down. And what's most interesting, after the treatment is removed, the biological age is still below the control. So you can achieve rejuvenation, which is once again very nice, because if you can achieve rejuvenation in a short treatment, you don't need to care as much for the safety. Once again, let's go back. Well, as I said, in mice, everything is nice and simple. You get whatever data you want, whichever omics you want. You have longitudinal data, you measure, you get something that exponentially explodes, and this is your biomarker of age. So in mice, it's sufficient to have a single number, which is the biological age, that you can devise drugs that actually lower it in a essentially persistent manner. So humans are different. It's not doesn't only mean that we do not have tails, but also if you apply the same program for humans, you will get surprising results. So first, and I will not give you many details on that, we have just published this work in Nature Communications. If you do something like that in humans and generate this kind of a single number, presumably a single number characterizing your biological aging, then as a function of age, instead of linearly or exponentially increasing as we age, that number on average in increasing in subsequent age cohorts would change ever so slightly. I mean, most of the change would be before the age of 20. And then after, let's say, age of 30 and 40, the number would increase a little bit, but the pace of that increase would have nothing to do with the exponential increase of mortality, as you would expect from all these incidents of chronic diseases and death. So essentially, you do not see, in mice, you see the exponent, in animals, in humans, you do not see exponents whatsoever. And the first part of this trajectory is just the growth. You can measure growth with whatever you want. You can do it with methylation, with blood formula, everything. But as we stop growing, so let's say, as you can see, the growth is separated from aging by the age of about 40 years old. And by the way, the mortality starts increasing at about 40 years old. But once again, you have no idea why we increase our chances to die or acquire a disease exponentially, and whereas our body composition, any measurement of your body does not have any exponential dependence. So in order to answer these questions, we built an app. This is not a consumer app, so we're not earning money with this app, but people are providing their measurements. Essentially, they provide steps per day, per minute during the day, or heart rate per minute during the day. We have tens of thousands of users, more than 100,000 users all over the world. We are working with big companies like PepsiCo, for example, on their projects, and with all kinds of other guys. The idea here is that by acquiring the motion data from people, this is not, of course, blood, this is not DNA methylation, but we have these numbers in tens of thousands of people longitudinally, because, for example, your health kit remembers your history of motion for a few years in health kit. So what we have found, not only we found, I mean, we did not found, as I told you, that we didn't found that people have exponentially increasing biological age, as we could expect. I mean, that was not true. What's most interesting was that in humans, remember in mice, the biological age was exponentially increasing till the moment when these animals are dead. In humans, we found that the biological age does not increase most of the time. Most of the time, the biological age in humans oscillates at around a certain equilibrium state. And I'm telling you that this would happen with any kind of biological age that you are using in humans. So when your biological age oscillates at around certain stable position, that means that contrary to mice, you are stable. You are not disintegrating all the time as these small animals do. That also means that once you are stable, it means that if I stress you, you would come back from your stress position back to the norm. And the time that is required to get back to the norm is another biomarker of aging. So once your biological age is not a good biomarker of age anymore, the dynamic markers are more important for those guys who are stable. So on this graph, we show you examples how we measure the recovery rate from fluctuations. And you can see that the decorrelation time actually increases in people who are elder, which means that elder people require more and more time to get back to the norm. So their resilience, their physiological resilience is actually decreasing as they age. So we have measured actually the recovery rate as a function of age. We used blood markers, we used physical activity markers from our app. It doesn't matter. Once again, all hallmarks of aging are correlated, which means it doesn't matter which biomarker of aging you are using for these studies. It doesn't matter what you do. You can find that in humans, as we age, our recovery rate is increasing, which means that our recovery time is increasing, which means that by reaching some age at around 100 years old, most probably you will end up in a situation when your body state cannot recover from stresses anymore, which means that any small perturbation would kill you, which means that it would be very hard to live more than 150 years old if you do not build interventions that target resilience rather than biological age. We were able to publish this work very well. Scientific American made an interview with us. So I think essentially we introduce a new angle at measuring the health states of human as we age. But we have not yet presented the answer. How it happens that although there are only linear changes to our biomarkers of age, where or how it happens that our chances to die actually increase exponentially as we grow older. So to understand this issue, we actually started looking at different people, let's say with magnification, and we found that the fraction, the number of people that are not able to recover from stress from the fluctuation analysis. Once again, to do that, you have to have longitudinal measurements, and we have them a lot from our application. So from our app, we found that the number of people who are not able to recover from stress actually increase exponentially as we age, and double every 8 years, as it should be. So in fact, it appears that humans exist in two forms. So most of the time, let's say 60 years of life, we exist like naked mole rats. So we're dynamically stable. It's very hard to kill us. So that's why we are living so long. And at late in life, some of us, well, eventually all of us, but at any given age, some of us acquire certain diseases that destroy our resilience, and we become unstable and start exponentially disintegrate. This is actually not a novel thing. It's just we were able to measure it with biomarkers as simple as biomarkers from wearable devices. But every doctor would tell you that, for example, the inverse hospitalization time increases, inverse hospitalization time decreases as we age linearly, and fraction of people who require long hospitalization time increases exponentially and doubles every 8 years, as it should be. So metaphorically, this can be described as a situation where we have a kind of potential, landscape, like in the mountains. So we are born somewhere high in these mountains, like, a mountain river. We are going down, unfortunately, because that's the shape of this potential. Most of the time we spend in the valley, and while we are in that valley, like from the right and the left chart, we are dynamically stable, which means that the perturbations do not lead to long-lasting effects. That's how we live most of our life. But as we age, as we go down in this valley, the height of the hill that protects us from the disintegration actually decreases, which means that we more and more often activate over the hill and end up in the abyss. And once we are thrown into that abyss, our biomarkers start deviating from the normal levels exponentially, and we quickly die. So which means that contrary to mice, where things are very simple, in humans, we are in a much more precarious situation. So our aging is not, in one-dimensional rollout, like in these little animals. In humans, and I think all doctors know that before talking to geoscientists, of course, that in humans, we are more like jumping between different metastable states that are characterized by different contexts of chronic diseases. And of course, as we jump more and more, our resilience is getting worse. And at some point late in life, we are suffering a catastrophic disintegration. So in order to understand the character of human aging, we have to have lots of longitudinal data. We have to use lots of AI. We have to build systems that do not attempt to describe our aging with the single number. So for example, dynamic models that presume multiple metastable states. We do that a lot. Other people are doing that as well. We see that human populations are very heterogeneous. We see different kinds of people that are characterized with different clusters of chronic diseases. Each such cluster would require different measures to intercept aging, to intercept further jumps. And of course, as we get more data, as we have more life histories of human beings, we will have more and more sensible description of this situation, and eventually I hope we'll find drugs that would not able to help us only late in life like senolytics, but most probably the drugs which would be able to keep which would help us to keep us young for long. So with that, I would essentially try to conclude with the idea that humans are not mice. Life in mice is simple. We can rejuvenate a mouse, no problem with that whatsoever. And in order to understand the character of aging in humans, we have to have more data, more sophisticated approaches, and hopefully with AI and common sense, we will be able to do that and produce therapeutics that can do a lot, which means that not only extend late life, to reduce late life mortality, but also I hope produce dramatic, probably fold improvement in human lifespan. Thank you very much.
Thank you so much, Peter. We couldn't be in more agreement with regards to the vast gulf between mice and men and mice and humans. So well said. I would now like to welcome to the floor our next speaker, Dr. Afshin Beheshti, a scientist at KBR at NASA Ames Research Center, who will deliver a talk entitled Mitochondrial Dysfunction as being a central driver for health risks associated with spaceflight. Afshin, it's a pleasure to have you back. The floor is yours.
So thanks for the introduction. And again, thanks for having me back. So this is going to be a little shift from the previous speakers, but it's going to have aging in there, of course, but it's going to talk about spaceflight, as Franco mentioned. So before I go on, I know probably most of you audience and then most of you here, the participants know about mitochondrial aging, but just a brief overview. We know that, mitochondria is heavily involved with the hallmarks of aging. I'm sure a lot of you are familiar with this paper that came out in Cell, when was this, back in 2013, called The Hallmarks of Aging. And it's a really nice paper talking about what occurs with aging and all the stuff that the previous speaker and other speakers have been talking about that occurs during aging molecularly. They really outline the steps there and what, this review goes over that. And of course, one of the key hallmarks is mitochondrial dysfunction. And as if you're not familiar with mitochondria is the powerhouse of the cells, basically your bioenergetics that keeps you going and keeps you active, keeps you healthy. And of course, if you have mitochondrial dysfunction occurring, this is a older review by Doug Wallace, who's one of the founders of mitochondrial genetics. He in this paper and on many papers after this, we're discussing, basically, if you look at your cells with age, the mitochondrial function goes down. And then this is related with like reactive oxygen species, oxidative stress, and eventually cell death, because, if your bioenergetics and your cells stop working, of course, everything else will shut down. So mitochondria is very important. This is like the overview, simplification of what mitochondria does, and, of course, and related with aging. But I'm sure people here know all about the real biological details of it, which will go in the context of spaceflight. And of course, every other disease, when you have mitochondrial dysfunction happening, it affects what you're doing. And with age, of course, with the decline you have with mitochondria, this is going to compound factors, and you'll have certain diseases related with it. So most people here, I'm not sure if you're familiar with spaceflight experiments and what data is used for that. So briefly, going over spaceflight in the space environment. And so the space environments involved with, there's many different things out there. So in Earth, we're, of course, protected. We got the ozone layer. And then outside the Earth, we have this magnetic field called the magnetosphere, or the Van Allen belt. And right now, all the experiments that are done in space, which is the low Earth orbit, as I said, protected by the Van Allen belt, is usually in that magnetosphere. So the International Space Station, the older shuttle missions that people have done, not just NASA, but around the world, all the other space agencies from JAXA to ESA and other ones around the world, Chinese. And so basically what happens in the Van Allen belt, you got a lower dose of radiation. So in space, you got several hazards. You got the space radiation, heavy ions, these heavy giant particles like iron particles, oxygen, silicon, from things that can happen from like supernovas and Earth are traveling through deep space. And you're exposed to that. And our magnetic field and our ozone layer on Earth, we're not exposed to that. We're protected by it on Earth. And then you got the lack of gravity, as you know. So the lack of gravity, of course, will impact many different biological functions in your body also. And then you got other factors here, as we highlight here. There's a review we had put out last year in Cell, talking about like the fundamentals of, or what we like to call the hallmarks, of space biology, where you got different things happening, like the distance, confinement, gravity, and radiation. But gravity and radiation is what I'm going to concentrate here on mainly, which impacts your health. So just give you an idea, like if you say go to a mission to Mars, which is like the future in a decade or so, a couple of decades. That's the plan, right? To go see what happens in Mars, colonate it, or have people actually fly there and see, explore it in person. So typical radiations you might get on Earth is like, chest X-ray and things like that are very low. And you got to keep in mind the type of radiation we get on Earth is very different than what we have in deep space with the heavy ions. On Earth, we have gamma. So gamma radiation is just like these fast sand particles going through your cells. And as opposed to deep space, is you got these bowling balls sitting and depositing massive amounts of energy in your cells. So the deep space mission to Mars is, relatively speaking, this is this much more radiation you might get compared to everything else, where there's the shuttle missions, or this is an older slide, so Skylab is now ISS, so the ISS missions. So that's a concern, right? A lot of radiation. How does that impact things? And then so the health risks that are involved is basically, females and males, there's a lot of similarities of different health risks you might get, but there are distinct health risks that are associated with female and male, we know. So things like cancer, sleep disorders, muscle degeneration, kidney-related issues, and a lot of things are correlated with aging also. So space, that's another concern in space. Does the space environment, meaning the radiation and the microgravity that's in space, would that, what optimal age group should be sent to space? Currently right now they're in the usually typical age of astronauts are between 30s to 50s. But should we send older people? What happens if, now that things are becoming more commercialized, as we hear with SpaceX and Blue Origin, Virgin Atlantic, sending normal civilians up to space that are not at the top 1% healthy population like the astronauts are, or the top 0.001%. Is that a concern? So we just heard recently William Shatner went to space, right? He's 90 years old. So with that age, would it be more beneficial, or than, let's say, sending a younger person, or would it not be, based on maybe acceleration of age due to the space environment? So the types of experiments that we do, just in case you're not familiar with, this is like we send up mice, for example, to the International Space Station. This is a video of mice that are on the International Space Station, our little mighty mousternauts, floating around in their cages. There's a paper we put out just describing the types of experiments that are done. And then there's, the ground experiments, the simulated space experiments we do on ground. For example, we go to a lab called Brookhaven National Lab, which is in Long Island, New York, which is a high energy physics collider. So NASA had set up a space radiation laboratory there in early 2000s, where you could take those heavy ions produced by these high energy physics colliders, and then take it like for example, an iron particle or a mixed beam of radiation, that'd be what you might get exposed to, let's say an hour in space, like iron particles, oxygen, and so on, and then put your samples in front of that beam and irradiate them. So that's some things that I've done and other folks have done who are principal investigators of grants, and they go here and run experiments. For microgravity simulation, like in animals and mice mainly, you have this setup, which is from my experiments, where you hang the mice by the tail, so it's kind of unloading the weight from the back of the leg. So it's kind of like the microgravity event. It's sort of silly, but it's the closest thing we can do to having a simulated microgravity experiment in mice. Another collaborator of mine at Beth Israel, Stuart Rucker and Mary McTrue, in the Boston area, they have these experiments called partial weight bearing rat model, where they have a harness and they could hang the rats at certain elevations to simulate like certain parts of microgravity, like 20%, meaning like the moon gravity or the Mars gravity and so on. So those are the type of experiments. And then let's say if you don't have grants, you're not running experiments, there's actually a treasure trove of data on NASA's GeneLab platform, which is here. This is the site where you can go and the data is publicly available, where it's all the bioinformatics, all the omics data, basically, genes, transcriptomics, genomics, any type of omics data that's really space is being collected and deposited here. And then GeneLab has made it where the hardcore computer scientists can get the raw data and play with it and come up with novel hypotheses and analysis. And then we also have provided in GeneLab like the higher order data, like the process data. So if you're not, don't want to spend time with starting the raw data, you can start with the process data. And plus, there's a whole visualization platform where people who are not as computationally inclined, they can actually go in the visualizer and type their favorite genes, let's say related to certain aging factors, and see how that might change in these animal models or cell models that are sent to space, or these simulated experiments with omics, and maybe have some novel hypothesis with age that like a previous speaker was showing, that could occur, that maybe space either accelerates or decelerates it. So now the actual story. So we had put out a paper last year that came out in Cell, where we found that mitochondrial stress was a central hub related to spaceflight that impacts the body. And the general overview of this is that, before I go on, I just want to, this is a work done by many different people. And in Gene Lab, we have these things called analysis working groups, which I run, that's open to the public. Anyone from the public can join. And I lead a multi-omics analysis working group where, anyone from around the world can join. We have many people there, and we work on, together as a collaborative open science kind of initiative projects that, could make an impact. So this project here was basically, I said, when people were involved with this, is there kind of a central switch or central driver in space that can impact your biology? Because in space you have a systemic effect. It's basically your whole body being exposed to radiation and microgravity. So the key is then, looking at Gene Lab, we utilize like already available omics data from mice, from like protein level, DNA level to RNA level, metabolites. And then, using our various skills in our group with pathway analysis, modeling, computational analysis, we started like picking out the story that came out. And the story came out over and over again, looking at many different tissues from mice that were there, and also cell culture experiments that were done previously, that mitochondrial pathways were always showing up were key. No matter, and we looked at certain different ages of mice too, no matter the age. And then we also had astronaut data. We had like the largest amount of astronaut data in this paper, physiological data from key targets with mitochondria that was related, to look at aspects of, in the human model, if it's related to that. And then also, you might have heard, I'll go briefly over this, the twin study data with Scott and Mark Kelly were in space for a year. Scott Kelly was identical twin to Mark, where Scott Kelly was in space for a year. Mark, his identical twin, was on Earth. So we have data from that, actually from the blood and RNA sequencing data to confirm what's happening. Granted, it's an N of 1, but it's kind of a proof of concept thing. So again, how does this relate with aging? Since this is an aging meeting, not a space biology meeting, is that again, mitochondrial being a key hallmark of aging. And what we put out as a review, mitochondrial based on this paper, again, is key hallmark in space biology. There's other factors that are related with space too, which I won't go into this talk, but there's others folks that are in the field that I know could give a talk about this. For example, telomere length. With aging within the astronauts that are in space, Susan Bailey, who's in Colorado State University of Colorado Boulder. I forget, I always mix those two up, but one of those two. But she's a very prominent PI in NASA World and Twin Study, and what she found is that, as we know, telomeres shorten length with age, right? So, but she found in astronauts when they're in space, starting with the twin study data, and now she has a lot of great data looking at further, the telomeres lengthened. So are people getting younger, or is there something else happening? But when they return, the telomeres get back to regular length. So with aging, there's some things happening that not only in the mitochondrial level, but other factors that we say are like epigenetic changes and so on. So specifically for the mitochondrial story, so we start out that we started looking at an unbiased way. Tell us what the data, omics data, tells from all this data that's available. And again, this is the cell data that we looked at, and the central overlapping pathways that, from all, long story short from this, to look at what was happening was all related mitochondrial activity and stress happening. And the majority of these mitochondrial pathways, which are in the green highlights, are downregulated. So it's heavily suppressed in the cells. Just the cell culture in space, coming back and then analyzing it, that was mitochondrial suppression key, in cells. And then once you start looking at the mouse data, these are from two different mice strains, as you see here, BALB-C and C57. And you got to just know one little distinction is that C57 mice tend to be more radio resistant, meaning resist to radiation. BALB-C mice tend to be more radio sensitive. So, that's a key difference. But even though that key difference is there are some mitochondrial expression differences that occur with them. But the overall story is that mitochondrial changes are pretty much universal. Depending on the organ, some organs might not be as prominent for one strain of mice for another, based on what you're seeing here. But long story short, here, like there's a lot of data here we did from RNA. This is a kind of a network level representation of mitochondrial activity in all these different organs: liver, kidney, eye, adrenal gland in these mice. And the inner ring is the protein data, proteomics. Outer rings is the transcriptomic data. And again, mitochondrial activity is really being dysregulated. This is the muscles. And the muscle it's going to be interesting how it happens with the key relationship to the liver. In general, the muscles are being downregulated in a lot of activity you see. And then this is just looking at mitochondrial genes overall, all the mitochondrial genes out there that are available from a source called Mitocarta, which is from Broad Institute, that you'd see that mapped onto the different organs. And you can see like certain organs, like here, the liver is having a prominent role. The liver here is having a prominent role in a lot of mitochondrial dysfunctions happening, which is, makes sense because that's also related to your metabolism and mitochondrial activity there. And it's all the organs have it. The muscle, interestingly, is the soleus muscle, which is here. And soleus muscle is one of the first muscles that really start degenerating in microgravity conditions. Going further, looking at specific mitochondrial factors, you can see like this is looking at metabolites happening during metabolomics in the muscle. You see all these key pathways, top pathways related to mitochondrial activity being dysregulated. Looking at specific oxidative phosphorylation genes, and then looking at integrated stress response genes, you see, depending on the organ, certain mitochondrial activity happening in a lot of things. And again, don't want to go too much detail, it's a limited time, but I'm going to show you the basic story. This is actually data from the twin study data. So Scott Kelly went to space, Mark Kelly was on Earth, the identical twins. And what we see here is the ground study, the ground twin, Mark Kelly's here, and the Scott's going to space over time. This is when they're during flight. This is when they come back. When it comes back from flight. And you can see during flight, these are mitochondrial DNA, or genes related to mitochondrial DNA, are being heavily dysregulated in space. Interestingly enough, that when you start looking at physiological data, this is compiled from 60 astronauts from the last few decades, related to mitochondrial activity, like antioxidant factors in the blood, or these factors related in the urine. When these factors in the urine are up, they indicate mitochondrial stress. And you can see this is before flight, this is returning after flight, 30 days after flight, this is during flight, 15 days during 30, 80, 180 days, so on. And same goes the same for all the x-axis. So you can see there's actually, antioxidant suppression happening. And then these factors here, like the PGF alpha, there it is, increased levels of OHDAG and prostaglandin F2 alpha as biomarkers for cellular oxidative stress happening, which is related to mitochondria. What's also interesting here is now we've done some metabolic flux analysis within the group, where you take the transcriptomic data and then you look at what the metabolic fluxes happen. Now this is starting to be the interesting interplay between the liver and the muscles. So the livers are in the mice are these green and blue. So green is mice that were up in space, and these are the mice that are on the ground. This is now the muscles, where there's the soleus muscle, or there's another muscle here is the EDL. Again, this would be the space flight is here, sorry, and then the ground flight's here, space flight and ground. And what we see here is that, interestingly enough, the ground is purple and the space flight's here. So what we see, interestingly enough, is this interplay where that, the liver is trying to be upregulated for mitochondrial function, while the muscle is degenerating and it has mitochondrial suppression. And in the literature, especially with aging literature, this interplay is seen where muscle cachexia happens with aging. And indeed, when people have studied mitochondrial functions in the liver, they try to overcompensate for this lack of dysfunction, try to get your metabolism up to hopefully overcompensate for this degeneration that's happening in your muscles. But in space, it doesn't seem to be enough to actually help degenerate. And then this is just interesting twin study data, again, looking at the RNA transcript with mitochondrial genes with different cell types. So these are different cell types that we're comparing in Scott Kelly versus Mark Kelly. And you can see that this distribution is going from pre-flight to ground versus coming back. And within time, you can see the mitochondrial genes are redistributing within these cells, which is pretty interesting. So now there's all these downstream impacts. So the functions of mitochondria, and there's a whole bunch of downstream impact on energy and immunity and so on, calcium balance. And one of the downstream impacts is the immune dysfunction. And what we see here is that clearly, like the immune dysregulation is being a lot of cases might be. Let me stay on this slide. Sorry. Is being suppressed. And what's interesting, when you look at the astronaut physiological data, for the sake of time, we'll just quickly skip to here because the transcriptomic data shows that you see that factors related to vitamin D or inflammatory factors are heavily are being expressed, overexpressed in the astronauts, while things that are related to vitamin D, which you want to have up, and other things are, your VEGF, meaning your angiogenesis, they're either up or down and being dysregulated. Again, so you go now in mitochondrial and fatty acids and your lipid, like homeostasis, is heavily correlated with each other. And this is just a review paper that discusses the nice one, talking about mitochondrial dysfunction and things that would cause for lipid dysregulation, things that could cause damage to your liver. For example, you could have non-fatty alcoholic liver disease, NAFLD, which would cause lipid accumulation, which we've shown in other papers that occur, which is downstream of what could happen mitochondrial dysfunction. And with aging, these things happen too. So what we see here, long story short, again, is that that the lipid dysfunction downstream of that, whether there's cholesterol levels in the astronauts, you see it coming up, which is a bad thing, or other fatty acid related things are happening within the transcript level. And what's interesting here now is, again, the metabolic flux analysis comparing the liver to the muscle. Same story as before, where in the muscle, there's heavy suppression with all these things related with either metabolism or glycolysis, things like lipid related factors are being heavily suppressed, while in the liver, they're being overexpressed, trying to overcompensate what's happening in the muscle. And then there's other factors related, like circadian rhythm, with sleep, again, with age, circadian rhythm issues happen. Space flight is a really common parallel to that, cell cycle and so on like that, and your cells are behaving and being greatly affected. And one interesting thing is your olfactory pathways are being heavily dysregulated in these mice. And astronauts have reported things talking about, their smell and taste kind of going a little haywire when they're on space and they come back and it recovers. So, there's different factors. And a lot of these things are actually downstream of mitochondrial dysfunction, or all of them are. So in reality, the summary of the work is that what's happening in space based on all this work is that we said, basically the key thing is mitochondrial dysfunction. And mitochondrial dysfunction is really upstream from a lot of these factors. And the parallel to aging is really interesting because, a lot of the factors I show, and especially when you see it, go back to, let's say, the hallmarks of aging paper, that occurs in space are similar. And now to figure out, let's say, is it beneficial to send someone at a certain age, because we already know with let's say when you're 70, you have a lot less bioenergetics and you have a lot more mitochondrial suppression happening as if you're 50 or 40. So is that going to contribute to further health risks? And just I'd like to make a little plug if you're interested in space biology research in general. We had organized, me and a couple other PIs, Chris Mason and Susan Bailey, had organized this large package of cell papers, of space biology papers that ended up in Cell Press. And we got these covers here that actually Scott Kelly provided. As you can see, the images are from the ISS. There's a 29 papers here, and you can go here and read all about space biology research. And the telomere work that I just mentioned, from Susan Bailey is also here that you can read about. And of course, the Gene Lab folks that are involved in general. So thanks for your attention, and happy to answer questions later.
Thanks so much, Afshin. A delightfully comprehensive and insightful presentation as always. So thank you so much for that. Viewers who have questions and comments for Afshin, please pop those in the Q&A box at the bottom of the screen, and we will address those during the panel discussion. And also, viewers may be interested to note that both myself and Deep Knowledge Group General Partner Dmitry Kaminskiy were co-authors on a paper with Afshin and over 30 other colleagues a few years ago, I think 2017 now, on the topic of optimizing human health for long duration space flight, colonization, etc. And that paper did briefly touch upon some convergences between detrimental health effects of spaceflight and the biology of aging proper. So for any viewers interested in viewing that, it's available on OncoTarget. Vive la radio résistance. So thank you, Afshin. I would now like to welcome to the floor our next speaker, Philippe Mertens, co-founder and CEO of Evocal Health, and he'll be speaking on unlocking the human voice as a biomarker for health and longevity. Philip, pleasure to have you with us. The floor is yours.
Thank you very much, Franco. Thank you very much in Deep Knowledge Group and Aging Analytics. It's great to have such a nice platform here, a platform for exchanging ideas, connecting with industry leaders across the world, and this in the field of longevity. It's great to be here. So I want to take you, or I want to invite you to join me on a journey, and a journey that we're on, that I'm personally on, with my company so far. It's the journey of unlocking the human voice as a biomarker for health. And as I know that the group is quite diverse and consisting of physicians, inventors, innovators, I will make it quite scientific and less practical. It's all about the practical application. I will dive with you into the why, the how, and the what. The why, so it's all about why the human voice as a biomarker is so important and so attractive. The how, I will dive deep with you into the topic, what's special about digital and voice biomarkers. And third, I will go into the what with you, how we develop a completely new kind of biomarker. So the journey we're on, it's the journey of longevity. And the journey begins with the birth and the end of the life. And there's the significant space in between. It's the journey in between. It's that what's consisting for longevity. It's extending human lifespan according to health span. And I have good news for you. We're united. We're united by the passion to extend the human health span according to lifespan, with technology and with science. And this conference is a landmark to show this. So what is your personal definition of longevity? When I ask this question on stage or in conference talks, I always hear different things. And I want to wrap up this whole thing in the beginning of my talk, just to allow anyone to follow me. So it's living longer while remaining healthy and vibrant. It's the space in between death and life. It's preventing diseases, so not getting ill, not getting sick. It's not dying, the most basic thing at all. It's looking and feeling younger. And for many people, it's growing biologically younger while growing chronologically older. And this is some kind of the secret to longevity. And when we look to the dimensions of longevity, it's all about first, prevention of premature death, second, life extension, and age reversal. And we have excellent talks so far heard at this conference. So what are biomarkers? I want to boil it down to make it compatible for everyone. A biomarker is a piece of anatomical, physiological, or biochemical data that is used to diagnose or develop a treatment plan for a patient. This is the official definition of the Food and Drug Administration, FDA. Now we're diving deep now in the field of digital biomarkers, and a digital biomarker is a biomarker that is developed from data. So we are looking at data that are analyzed using advanced analytics, AI, and we extract previously invisible insights. So you imagine, or you remember this slide. It's the importance of digital biomarkers. There is the end and there is the birth of life. And when it comes to the space in between, I have visualized here dots, and every dot you can see here is 1 day in the life, 1 year of a patient. There are these red dots. These are dots where symptoms worsen. The patient that is sick, that has an illness, has a worsening of symptoms. The blue ones are naturally those days where the patient seeks for the clinician, or when the patient who is in a clinical trial is reported to the clinic. And you see there's pretty much gray or white space in between. These are all days there where we have no clinical picture of the patient, where we have no data so far. And this is where digital biomarkers come into place. So the question for us was, what if we could early detect and monitor diseases with nothing more than our voice and a smartphone, using the power of AI and data analytics? And this is what we're on with Evocal Health so far. We're unlocking the human voice as a biomarker for health. And we're a digital diagnostics company based in Germany. During the past years, we have developed a device agnostic platform that enables us to collect, monitor, and then analyze voice data for self-management, health monitoring, and decentralized clinical trials. It's a biomarker that is quite special. It's non-invasive, it's cloud-based, and the whole suite is privacy compliant. So boiling it down, the voice as a biomarker is a brand new way to measure and monitor our health every day. They deliver, and we have heard very interesting talks so far, objective and unbiased data in the home care environment, which makes it quite interesting for remote patient monitoring and decentralized clinical trials. These deliver non-invasive insights, so we do not have the patient having much of involvement. So we are a biomarker that can be used with minimum patient involvement. And this is what makes the human voice as a biomarker quite special. So digital biomarkers are changing the way we understand diseases. giving a more precise picture of a patient's disease progression and his or her response to a certain treatment or drug. The mission behind, we don't want to have this gray space here where we do not have data. We want to remote patient monitor the patient. So let's come to the voice as a biomarker. We're currently in a very interesting time. We have voice interfaces all over. Big tech and user demands are speeding up. The convenience and conversational AIs are something that is quite mainstream so far. COVID-19, we have heard this example so far, has accelerated the demand for digital health, and we're currently actively employed by health authorities. There is this longevity economy, what this conference is all about. People aged 50 plus, especially here in Germany and the Western world, will compromise 41% of the population, tripling their GDP impact by 2050. And there is this big topic of value-based healthcare. The global healthcare system is currently shifting to evidence-based medicine, patient-centered care, delocalization, and cost effectiveness. And all of this is for us the sign that there is a big need. There's a big need for fast, non-invasive, and cost-effective new biomarkers. I want to now dive with you into the mode of action. So you might ask yourself, what is the medical ratio behind? Why is the human voice an interesting biomarker? And let's dive deep into this topic now. So the human voice, many people don't know this, is one of the most complex systems of the human body, and it's a powerful biomarker. When the human voice is produced, there is a very large set of tasks for the coordination of different organs, muscles, and sensory nerves that needs to be coordinated by the brain. And during the past years, we have shown that diseases significantly impact the physiology of major sound-producing systems. And changes in the acoustic or semantic parameters that we detect from the human voice are resulting in very disease-specific changes. You might know the situation when you talk to relatives, to your mother, to your partner, whoever. You hear them. When you talk to them, you hear that something is not going on. When I talk to my grandma, the first thing that she always reminds me to is, have you got enough sleep? Are you eating well? Or are you physically engaging enough? And this is what science has shown, that there is a medical ratio behind. So there's decade-long clinical research behind the human voice as a biomarker. It's showing that there are more than 3 promising indication fields. We have now more than 20 years of fundamental scientific research behind. There are more than 150 core publications about the human voice as a biomarker. And we deeply believe that the opportunity to unlock this interesting field is now using AI and ML. My co-founder Dirk has for more than 25 years started to discover the potential of the human voice in his early studies. And now with the very powerful smartphone devices and microphones in our pockets, it's the right time to unlock it and bring it mainstream and to the patient. I want to give you a little bit of an introduction into how our technology really works and what's the mode of action behind. So boiling it down, making it quite easy, there are 4 steps that we undertake when analyzing the human voice. At first, we're digitizing and normalizing voice samples. Then we correlate or we detect different parameters in the human voice, 150 to 200 parameters that the voice is compromised of. And then our pattern recognition, AI and ML models come into place. And in the end, it's all about detection of correlations and causalities to indications using EPROs, evidence-based guidelines, and especially the clinical pictures that comes from EHRs and so far. The process of identifying clinical grade, and this is important to mention here, we do not talk about esoteric or other stuff. It's about clinical grade vocal biomarkers that are validated, that are medically validated in RCTs. And when we go into the process, there are 4 main parts. First, it's the part of data collection. We collect data, so we collect voice recordings with our cloud-based solution. And we always need to have clinical data that we can compromise and that we can label our data sets with. The second is the data processing part. We normalize everything. If it's the noise behind, if it's the phone ringing, or if it's you standing in the traffic jam. We extract parameters and then we have very nice labeled data sets. And then this is the important part when our data analytics and our biostatistical analysis come into place. We train the ML models, we identify candidates as in every drug development process. It's always the same here in the vocal biomarker detection process. And then we validate and replicate with internal validation test sets and external validation test sets. Listening to this talk now, you might ask yourself, where's the technology standing? What are technology readiness levels? And I always like to emphasize this in the 4 horizons of focal biomarker innovation. You are probably all familiar with this whole concept of the horizons of innovation. And we have put this into a very nice, how I think, visualization here. So the first stage, the first horizon was digital health management, having the patient being in his or her baseline. We were quite easily able to position the patient in his or her disease progression, having his or her baseline data. Horizon 2 is when digital therapeutics or self-management devices come into place. Having Horizon 1 technology allows us to have longitudinal data and to monitor the patient as a digital therapeutic and in self-management cases. Horizon 3 is all about bringing the diagnostic component in it, having very specific models, having very sensitive technology. And this is where we all aiming for. This is what this conference is all about. It's prediction. Our big mission is to go into prediction with the technology within 3 to 5 years. And predicting health, I think, is one of the most interesting things to unlock the human voice as a biomarker for health. So I went a little bit on the gas pedal here because I know that the conference was quite out of schedule. I hope that you have questions. I always invite any one of you to join me to connect via LinkedIn or follow our company on LinkedIn. You see here the barcodes that you can scan. And yeah, I was very happy to talk to you. I'm excited for the further talks now to listen to. And yeah, thank you, Franco.
Well, the pleasure and the excitement is all ours, Philip. So thank you so much for that. We have a great deal of respect and prioritization for innovative modes of biomarkers. I know that our next speaker, Anastasia, has also done a lot of work on image and photograph-based biomarkers of aging. So I think you'll be keenly interested in this next talk as well. A reminder also to all of our viewers who might have questions for Philip to post them into the Q&A box at the bottom of the screen, and we'll do our best to address them during the panel discussion. I'd now like to welcome to the floor our next speaker, Anastasia 𝐁𝐢𝐨𝐬𝐞𝐫𝐤, CEO and co-founder of Hot AI, who will be unveiling some recent developments on the use of artificial intelligence to confirm rejuvenation treatment success, with a special highlight on the role of gender-specific scales in performance evaluation. Anastasia, great to see you again. Great to have you here. The floor is yours.
Hello, Franco. Hello, everyone. Great pleasure for me to attend this conference. Last day was amazing. Today we heard a lot about different practical applications, which is, I think, very exciting, seeing that advances are actually coming into life. So today I'd like to talk about biomarkers that are based on imaging data. As mentioned in the title of my presentation, specifically I want to talk about the gender-specific scales. So when it comes to imaging and the way they look, traditionally it is believed that this is something more related to their female sex, so that women are most interested in rejuvenation interventions to help them look younger, which I want to assure is not 100% true. So in recent years, we see a big rise in their beauty tech products for men. We're seeing rise of different skincare brands, interventions, and men also want to be not only healthier, but we also want to look younger, being associated with a younger group of individuals. And I think that this should be praised. And we ultimately need to identify these visual biomarkers but will help us look younger, because it will have a very big impact on the mental health and the way we make our decisions, and pretty much contribute overall to our satisfaction with our life. So when it comes to the anti-aging, let me talk a little bit about the motivation. So for some of the people, that's primarily slowing down the aging process. So if we look deeper into what is actually slowing down aging, and we will not now think specifically about the different mechanisms, just something that usually a consumer or a patient would want to have is staying healthy, so not experiencing deteriorations of cognitive functions, and in the end also being perceived as an individual belonging to a younger age group. Then if we want to achieve this anti-aging effect, we can break down our to-do list into 3 parts. So we can think of different interventions. We can try to identify the very nature of the aging process and try to fix the broken mechanisms. And in the end, we also might think of modifying biological processes which are not normal, not like broken. They're considered to be absolutely normal, but in the end, they lead to the deterioration of our function. So as I think most attendants of this conference know, there is no agreement in the aging community of what are the reasons of the aging. So there are a couple of different theories. And I think in some of the previous talk we had at this conference, it was highlighted that even different aging clocks might not be in accordance and may not be correlated, but it's absolutely fine because it's likely that we're indicating and taking into consideration the different aspects of aging, and aging it seems to me, and that that many people share my opinion, it's multifaceted process. So when we would like to measure the actual anti-aging effect as a consumer, as a patient, we would like to see some changes. So that is the reason why nowadays more and more different aging clock appear. And some of this, if we'd like to structure and assign them categories in terms of, are these clocks more oriented to the patient or are they more oriented to the R&D? We can build let us call it heat map, and assign some clocks that were more R&D friendly and some clocks will be more patient friendly. So when it comes to more R&D friendly, or like better to be utilized in R&D practice clocks where there's a clinical parameters change, then we can also measure the effect of anti-aging interventions in some model organisms and see if they have longer lifespan. So for example, rats or other model organisms. If we would like to shift, closer to the patient, then we can use different cognitive tests, we can use functional tests, we can use self-observations. But if we want to make the measuring of the effect really explainable, this is the time when we need aging clocks. And aging clocks, again, can be utilizing different tests or like frailty indexes, or they can use different omics data, but we also now start to appreciate their photographic clocks, primarily because they're available, you can use image data, they're non-invasive, they have multiple benefits, and this is something that you can take on a daily basis and see how your features change over time. Again, I'd like to highlight here our partners from Deep Longevity, with whom we collaborate very closely on understanding how different clocks correlate with each other. So our role here as 𝐁𝐢𝐸 and our group primarily works on photo aging clocks, but we're also having a couple of research to understand how these clocks correlate with your behavior age, medical history age, microbiome, and see if we can identify a couple of trends. Again, before we didn't see that necessarily all aging clocks have to be correlated. Then let us talk more now about skin. So if we are having a biomarker, let's say photographic biomarker of using skin data, everyone would like to understand how they compare to others. So first, aging clocks is something that help us predict our age. We have to describe our aging process, but at the same time, we also want to know if I'm doing great compared to my ethnic group, compared to my age group, and compared to my gender group, just because I want to not be compared, population in general, but I'd likely want to be compared with a group with whom I share more similarity. And to make this skin and visual evaluations, there exist a couple of methods. So most of them are utilized in the lab practice. So we can divide them into 4 groups. These are different sensors that measure time series. If you know, look into what's the type of data they produce. Then there are different biophysical measurements, like different spectroscopies. They also produce the time series. Then we have a clinical evaluation. So clinical evaluation, then an image is presented to an expert, and an expert will be producing a grading according to some parameters. Then we have imaging methods that utilize pictures, and these are the methods that usually automated. We use computer vision methods. So this is some standard in the lab. Important to mention, their data standardization is really important here. So when it comes to translation from the lab to the consumer, we always have to keep in mind that accurate prediction is only available for standardized data, and to the extent possible in the real utilization of photographic clocks or skin data, it's important that data is very standardized. Otherwise, we're not able to produce meaningful results, and all of these types of data are not very robust when it comes to the conditions of how this data was captured or to change of the sensors. So in that sense, it applies a particular limitations to the use of photographic or other skin data. So in our research, we utilize all of these methods, but for their solution oriented to the patient, we decided to stop only on the imaging methods for 2 main reasons. So first, the data for images is highly available, so it's massive. You can now capture with your mobile phone, and compared to other data types, it's in fact less noisy when it is well standardized. And the last argument for us was that you don't need any special devices to capture this data. So what our technology has inside is, first, as I said, standardization is key when it comes to image assessment. So we have software that utilizes a combination of classical computer vision methods and neural networks to allow a patient or a consumer of longevity services to capture a very standardized, well-lit, high-resolution picture. Then we're using image quality additional algorithms already on the cloud side. So this library that is highlighted in the left is utilizing just resources of their smartphone or iPad. So it makes it very effective, and you don't utilize a lot of traffic. So you just capture a good picture using this library, and then you will be sending it to the cloud. So this way we can dramatically improve the rate of quality of the data, which is of course important to make extract any insights from the data. So after that, the data is standardized. We eliminate all pixels that are not related to skin. The primary reason for that is neural networks might take into consideration features that are not associated with aging. For example, your hair, the background, the eyebrows colors. So to limit the impact of environment or any style preferences, we just use a so-called skin atlas. And lastly, we predict a couple of skin metrics, which we will be analyzing in our research. So this is, for example, of how skin atlas looks like. We identified all features related to skin, then we break down the face into different facial zones, and we will be predicting parameters separately for every facial zone. A very important question is how inclusive is the technology? I think this is a question that we are hearing more and more nowadays when it comes to AI analysis. To address that, our processing includes decomposing of a picture into different spectral channels so that we can analyze image channel related to different facial parts and in fact molecules. So we can differentiate channel which is related to hemoglobin, we can differentiate channel which is related to melanin, and this breaking down picture into different channel allows us to process every channel separately and therefore extract features equally well for a fairer skin or melanin rich skin. So here we can see an example of how the technology, for example, can sort skin according to the uniformness of skin. And you see that in the class 1, we have very uniform skin. In the class 9, we have very ununiform skin. And the neural network model is not discriminating skin according to their level of melanin. As said, we anonymized all the pictures, then we processed them with about 14 different metrics utilized in the study. Not all of these metrics were analyzed further, but we picked up just a couple of them. And then every metric had a score where 0 was the lowest score and 100 was the highest score. So the motivation for our research was to see, will we have same results for males and females, and how their skin metrics will be distributed within different age groups to identify the visual markers that changed most significantly with age, and the ones that we can call the potential biomarkers of aging. So of course, a good biomarker needs to show correlation with mortality and lifespan. So this is basically the second step to show how good is the biomarker, but initially we need to understand what are the candidate biomarker. For every face picture that we had in our study, we identified every parameter independently for every facial zone, and then they used a nonlinear formula to produce an overall face score, and the coefficients for this nonlinear transformation of facial area scores were identified depending on the nature of the perimeter. For example, if we talk about redness, we would give higher scores to cheek areas because there are more they have more blood vessels, and this is why redness is more prominent on the cheeks than on the forehead. So every time the feature was engineered individually, depending on their biological nature of every parameter and how and the biology that impacts the change of this parameter. So here are the results. You see, this is a big graphic. We selected 10 skin features, and we analyzed the distribution for 6 groups. So we had 6 groups. We took females and males age under 30 years old, between 30 to 45 years old, and older than 45 years old. This split was driven by their data set. So we wanted to have same number of subjects within every age group for males and females. So then we calculated the facial parameters, and we built histograms to see how these parameters were distributed within different age groups. So our assumption was that all of our skin metrics, our skin quality, should decline as we age. Generally, we wanted to know if it's true or not. It's true. So we see that our skin metrics decline as we age, and there was none of the metric that had, its best parameter when you are young. So then, I'm sorry, I think that I have some trouble with the slides. I hope you see the right one. Then we were assuming that so we were thinking that all parameters decline as we age. In fact, we saw some decline with age, but the results were not same for different parameters. For example, for sagging, we identified that it's true. And in different age groups, the parameters were distributed not equally. And for both males and females were deteriorated with age. Surprisingly, dark circles didn't show that much dependency with age. We saw that for eye bags, wrinkles, and pores, they indeed deteriorated with the age significantly. Some parameters showed the difference for the gender. So we saw that for uniformness and pigmentation, the decline was more for male group than to the female group. And we might say that these are indeed the need for us to build separate scales for males and females because their very nature of male and female skin, which is impacted by hormones and the structure, will be different. And if we want to compare the improvements or deterioration for males and females, the scales indeed should be calibrated for every respective gender. In our research, acne was excluded from this comparison just because acne is usually attributed to the younger age, and it is quite hard to find enough samples of, let's say, subjects who are affected by acne at a more mature age. So to conclude, in our research, we showed that computer vision methods can be applied to process selfies, so not only lab data. So we can make clear separation between different individuals and their parameters. We saw that this approach will require a lot of standardization at software level and also at the post-processing stage. And we identified that we can process pictures for different genders, for both males and females. However, not all features are reliable to be used as biomarkers of aging. So we identified that wrinkles and pores are the best features to evaluate their skin aging. And we also saw that sagging and eye bags are the second tier of these features that can be utilized for tracking the aging process of our facial skin. I would like to thank you for your attention. I'm really happy to collaborate. If you would like to use our aging clocks, if you would like to use our face aging features and face parameters in your research, please contact us, and we will be happy to make research with you or any commercial partnerships. Thank you.
Thank you so much, Anastasia. There's so many developments included in your presentation. Since I first became aware of this project around 4 or 5 years ago through your 𝐁𝐢𝐨𝐬𝐚𝐫𝐤𝐞𝐫𝐬 project. So very exciting and very interesting for me personally. So thank you.
Thank you, Franco.
I'd now like to welcome to the floor our next speaker, 𝐁𝐢𝐨𝐬𝐚, CEO of the DNA Company, who will be speaking on the topic of hacking your DNA to reverse chronic disease and slowing aging. It's a pleasure to have you with us today. The floor is yours.
Awesome. Hey guys, so really what we're going to look at is this new sort of world in genetics that's crossing over from hardcore genetics, meaning that this gene means this gene means that, and really only applying to genetic conditions and moving away from that to chronic disease. I'm going to show you a report that looks at one simple element, and we're going to talk about, I'll use breast cancer as an example. We all hear about the BRCA gene, right? We know that that's a scary thing to hear about from women. And we even have women that hear about getting a report about a certain version of the BRCA gene for which they would then go get a mastectomy to be preventative. So this is what we're saying. How do you go from genetics, meaning that a genetic condition that's directly tied or correlated to functional genomics, as why does the disease happen? You know, in the United States, 90%, I'm sure someone's spoken of this already, of the healthcare budget is spent on chronic disease, most of which is preventable. So how do we prevent? How do we use genetics to prevent? So there's looking at, again, the BRCA gene equals breast cancer. Let's prevent by mastectomy or staying ahead with regular mammograms and checking. Or there's what is going on in biochemistry that would actually lead to the breast cancer, because BRCA doesn't cause breast cancer. BRCA is a gene editing gene that fixes other and repairs other genes. And if it's failing, it's not doing its job of repairing, but something upstream or downstream is actually causing the problem. So we'll use that scenario as an example of, well, what causes the problem? We know that most women, you know, get breast cancer in and around the menopause age. That's the most common age. And if we look at this map, we look at the sexual, hormone pathway. How do you go from progesterone to testosterone to estrogen, which is how you produce and clear hormones? In that process, there's a gene called CYP17A1, which can determine how quickly red light, very slow, yellow light, medium, green light, very fast, convert those progesterones into testosterone. And whether you're a man or a woman, you do the same thing. A man does it daily, a woman does it monthly in her monthly cycle. That's the difference. Once you've created these testosterones, you either clear them, you get rid of them, you convert some of them into DHT, which is that manly man version of testosterone. You have the ripped, striated muscle, but it also may affect your hair follicles, make you go bald. or you convert it into estrogen. So we have some women that we see at a very quick pace, green light, convert their progesterone into testosterone, so that initial pool of hormones is very full. These lights are red, so they don't go in either direction, and there's a green light here, so a lot of estrogen, right? They're estrogen dominant is what we call them. From there, you now don't clear the estrogen itself as in your menstrual cycle. You first convert it into a metabolite, 2, 4, or 16 hydroxyestrogen, one of the three. 2 is very healthy and clean. You want that. That's what a woman would prefer to have. 4 and 16 are highly toxic. So if you're producing a lot of estrogen and then you're converting into one of these before clearing, you're creating this toxic metabolite, right? So we're now looking from one gene means this to we're starting to paint the picture, the story of what is actually happening in biochemistry. This is the functional genomics versus genetics. This gene means this gene means that, versus how does the body work and what gene instructs each step. So now we've gotten to, you've now perhaps produced a toxic metabolite, 4 or 16, right? We then look at, well, how do you clear them? You know, if we know that, you're doing a good job, a poor job in what metabolite you're creating, now this toxic insult is there causing damage to the cells. How well do you get rid of them? These are the genes, COMT, GSTT, GSTP, SOD2, that get rid of those toxic metabolites. You may have a medium version, you're doing an okay job. You may have green, which is not, you're doing a really good job, or these could all be red lights, which you would be surprised. It's not a 2 or 3% problem. This is like a 25 to 30% problem where people don't clear the toxic metabolites that well. In fact, this gene here, GSTT1, forget about a variant or a version. You might not even have the gene. It's possible to have what's called a copy number variation, meaning it's not just about a SNP or variant, but you didn't actually get the gene, so that instruction isn't there and you don't do this step of the process. Now, if we go back to, I'm estrogen dominant, I produce a toxic metabolite, and I don't get rid of it. We've now looked at the entire cascade from beginning to end. Forget about BRCA and forget about a single gene that points to a 80% chance of likelihood of something, but you don't know, am I 80 or 20? You have to also study the 20%. Why did they not get the disease? Then you have to study the 80% to understand why did they get it. So in this scenario, if I produce the high, if I'm first of all estrogen dominant with a lot of estrogen, producing a toxic metabolite and not clearing it, and I'm menopausal, which is when most breast cancer cases happen, well, guess what? When I'm menopausal, I no longer have a menstrual cycle. These toxic metabolites that I'm not clearing that well, but still producing, where do they go? The body stores them in fatty tissue. Where do women have fatty tissue? In the breasts, in the hips. What's in the breast that was never meant to deal with this level of toxicity if you have the 4 or 16 hydroxy? It's all that glandular, like all the glands and everything that's going on, that literally the cellular structure was not designed. The lungs, the gut, first line of defense. They were designed to deal with that kind of toxic insult and actually clear and filter and send things to the liver to metabolize. Breast? No, it wasn't. And this is why you have all of a sudden 85% of North American women, at least at some point or for a long period, on a birth control pill, which adds to that estrogen pool, which adds to that metabolite that is difficult to clear. How many more women that get past sort of the fertility age into menopausal age now start taking hormone replacement therapy without understanding their cascade, and again adding more to this pool, which adds more to this pool, which then becomes hard to clear. That toxic insult becomes exaggerated. How many mimics do we have? How many foods do we eat that are estrogen dominant, and again add to this pool, which adds to this pool, which becomes hard to clear? So this is one example of how you take a simple disease, I shouldn't call it simple, but something that we treat as very simple, saying, hey, genetically, if you have the BRCA gene, you have a higher propensity for breast cancer, and that scares women. Versus, why does breast cancer happen? Let's not bring it down to the simplicity of gene equals disease, which is the case for things like sickle cell syndromes, non-Hodgkin's lymphoma. These are genetic conditions where a genetic switch needs to be turned on or off, and there's companies out there that are doing really great work to create genetic therapeutics to turn those switches on or off, right? But that's this much of healthcare. 90% of we said is chronic disease. You're not born with type 2 diabetes. You're not born with cholesterol problems. You're not born with breast cancer. Why does most disease happen in and around the age of 50? And then by the age of 60, the average American has 2. Because it takes that long of mismatching your habits, your environment, nutrition, lifestyle habits, those loads that you're putting on your cells that you're genetically were not designed to deal with, to lead to the cellular inflammation that leads to the disease eventually. So this is what we've been working on, and this applies, you name a chronic condition, you can think at this level. So you can think beyond the symptom masking. You can think beyond cholesterolemia is a disease that we need to treat with statin or Lipitor to get rid of the cholesterol. Why is the cholesterolemia happening? It's inflammation in the endothelial lining, which can be predetermined genetically. It's the inability to clear the free radicals and oxidative stress that causes that inflammation that then leads to cholesterol being deployed to reduce the inflammation. So you can test a 5-year-old child and understand that this is their hormonal pathway, this is their cardiovascular pathway, this is their detox, methylation, anti-inflammatory pathway, and start to predict that if these are your environment, nutrition, and lifestyle choices, you're likely to get ABC disease. But guess what? As we always said, this is an 80% likelihood, which means 20% didn't get it. So let's also look at what their habits are. The same suboptimal profile, but they didn't get the disease. So what were they doing right? And that's an area we ignore. And if we start to focus on that and bring and draw in more of those right choices and plug them in, we start to prevent and reverse disease. And in our research, we've done this. We've taken people with chronic conditions like cholesterolemia, like breast cancer, like diabetes, and not only treated, but reverse the conditions. We've seen things coming in people and help prevent. So how do you now translate this into anti-aging? It starts up here. The same way you can determine the cellular structure of all the various components that make us and understand how prone they are to disease. You can also understand the neural wiring or your neurochemicals based on your genetics, meaning when it comes to dopamine, pleasure and reward, how do I experience pleasure? Am I entrepreneurial? Am I more laid back and not seeking reward? When it comes to noradrenaline, am I susceptible to PTSD and trauma and fear? Or am I more going to surge ahead? When it comes to serotonin, your ability to regulate your mood and deal with anxiety, and deal with constant irritability, how do you deal with those things? When it comes to your mood in general, brain-derived neurotropic factor, and your ability to develop new skills, and your ability to cope with traumas like a legal letter in the mail. We start there when we look at anti-aging, and if we understand the human brain, you can then understand how you perceive everything around you. What would cause the stress and the cortisol? What would cause you to go in this direction or that direction? Then we bring in cellular structure. And we start to say, again, taking this functional approach, no single gene that is the anti-aging switch, but how do you deal with oxidative stress? What are you doing in terms of the SOD2 gene, and how well does your mitochondria actually clear oxidation, which would then lead to cellular stress, which would lead to that rapid aging, the faster wrinkles, the hair going white earlier, because you're not, again, dealing with that load or incapable because of your genetics. What would then lead to the ability for you to have sort of better bone health and your vitamin D cascade? It's not as simple as taking a blood test and saying, yeah, you have enough vitamin D in the blood, but guess what? That's step one. You also need to transport the vitamin D to the cell where it's actually used. There's a different gene. You may have the best gene to take vitamin D out of the sun and out of food, put it in the blood, but do you actually transport and utilize it? Then there's another gene to actually bind it at the cell. Now that you've got it there, you actually have to use it. And for some people for whom you test their blood and say, yeah, you have enough vitamin D, bone health should not be a problem. But meanwhile, there's an anomaly of they still ended up with osteoporosis. There's still a problem. That could all be prevented if you understood the full cascade as opposed to gene siloed or segmented approaches. You can also look at the FOX3 gene, a simple gene that looks at how do you deal with breaking down the proteins that would lead to rapid aging, that can cross the blood-brain barrier, that can cause things like Alzheimer's and dementia. Is your body metabolizing those things, or are they free-flowing and crossing that blood-brain barrier? So there's a lot of things we can look at, and it starts with first understanding biochemistry. What does the body actually do? Then instead of looking at genetics in the context of genetic conditions, meaning this gene means this, how do you then mirror and match the biochemistry to understand the full cascade for each process? From there, you can then start to understand what are the environment, nutrition, lifestyle loads that would impact that cellular cascade that would lead to a suboptimal outcome or a positive outcome. And from there, you can start to manage chronic disease and aging from a preventative or disease reversal perspective, because you're now dealing with a root cause, several layers before the symptom. And this is why, as I close, why we call these things chronic disease, because they're not acute in nature where it started today. The thing you're experiencing started 7, 10, 15 years ago when you started to make the wrong choices. That cellular load that eventually leads to inflammatory level where you can't handle it and expresses as disease takes that long to achieve. So this is how we say that you can dive into genetics a little bit more, understand it functionally, be able to treat chronic conditions, but also look at it in terms of how do you apply this to aging and slow that aging process down.
Thank you so much, 𝐁𝐸𝐸. Very interesting. For those of you in attendance today who have any questions about 𝐁𝐸𝐸 or the DNA company, please pop those into the Q&A box below, and we'll do our best to address as many as possible during the panel discussion a little bit later today. Thank you once again, 𝐁𝐸𝐸. It's a pleasure.
Thank you.
I would now like to welcome to the floor our next speaker, Dr. 𝐭𝐲 𝐿, founder and CEO of 𝐁infinity Precision Medicine, who will be speaking to us about personalized optimization of NAD and health via laboratory testing. Dr. 𝐭𝐲, very nice to have you back with us today. The floor is yours.
Thank you very much, Franco. And so I'm going to give a non-academic, non-professional talk today because I just quit my professor position. So I can spend more time with my company, 𝐁infinity Precision Medicine. In 𝐁infinity, we develop innovative biomarkers that are actionable. So our customers can use the information to optimize the health and improve their healthy longevity. Today, I'm going to focus on one of the biomarkers tests that we have developed in 𝐁infinity. I hope at the end of the talk, you will agree with me that NAD is probably one of the most important, if not the most important molecules in terms of managing health and longevity. So what is NAD? Well, the molecular structure is shown here on the slide. It's a small molecule, very simple, but it has many, many different functions because NAD is a coenzyme that's essential to life and is found in every single cell of living things. The enzyme is very important, or the coenzyme is very important for the function of over 400 enzymes. You can imagine why this molecule may play a very important function in everything in life. And these enzymes include some of the very important ones that are very important for longevity. These are the 𝐁𝐸𝐴𝑛𝑧, the PARPs, and the CD38, and so on. So what are the most important functions of NAD? Most people know that NAD facilitates the hydrogen transfer. You can see the two forms of NAD, NAD plus and NADH on the right of this slide. And the only difference is the hydrogen on these two molecules. So the hydrogen transfer is very important for the production of cellular energy in terms of ATP. And this is required for several of the energy production processes, including glycolysis, the Krebs cycles, and oxidative phosphorylation. So without NAD, you wouldn't be able to produce ATP, and we wouldn't live for very, very long. Some people believe we may not live longer than a few seconds, but maybe at least we will not be able to live for very, very long if we don't have NAD in our cells. If you look at the hallmarks of aging that many people talk about, I'm not really a big fan of the hallmarks because I think it's a good starting point. It does not address all the issues. But if you look at the 9 best known hallmarks of aging, almost every single one involves NAD. And because over 400 enzymes are involved when require NAD for their function. So you can imagine many of these important biological processes do require NAD to function properly. The most important ones are listed on the left, and NAD is involved in DNA damage repair, in oxidative stress, in inflammation, cellular senescence, nutrient sensing. It's important for supporting immune health, to protect the brain function, improve glucose intolerance, insulin sensitivity, and on. So you can read the slide probably faster than I can read. The bottom line is NAD plays such an important role in all biological processes, and therefore it's also very important for anti-aging or to improve our longevity. The second main reason why NAD is so important is that NAD is believed to decline with age, although the data are not very abundant in the literature. On the right side, I have shown you some data that are generated in Gfinity, where you can see the level of intracellular NAD on the y-axis, and on the x-axis you see different age groups. I actually cannot see the right one is hidden by the picture. So if you look at the very young adolescents from 10 to 20 years of age, the 25th percentile of intracellular NAD level is around 34 micromole. And that goes down to about 26, 27 micromole in the 20 to 30 years old. And when you reach 30 to 35 years, that goes down to about 22 micromole. And when you go to the above the 50s, I cannot see the number on my screen. It's probably around 20 micromole or so, maybe 22. So these data are from hundreds of individuals that we have analyzed. And this is I believe, the best evidence showing an age-dependent decline of NAD in the human population. Now, this decline in NAD is also associated with the loss of function and the vitality, and is related to many age-related diseases. Many of these issues are mainly related to the sirtuins that require NAD for their activation. So there are two different types of NAD that you may want to be aware of. And if you look at the location of the NAD molecule, and so most people talk about NAD that's inside of the cells when they talk about NAD. And this is what I call intracellular NAD. We actually trademarked this term. And intracellular NAD is found inside of the cells. The level is relatively high. And in most people, it's between 20 to 50 micromole. I have a typo here. And the most of the functions we'll talk about in the previous slides are attributable to the intracellular NAD, because that's what has been studied the most. But there is also NAD that's secreting from inside of the cells to the circulation, into the plasma. And this is what I call the circulating NAD. Circulating NAD is not very often talked about, and you may or may not have heard of it, but these NAD molecules found in the plasma circulate all over the body. They play potentially very important roles because NAD can serve as a ligand, and they are NAD receptors on certain cell types, especially neuronal cells and the immune cells. So as a ligand, NAD can bind to its receptor on the cell surface and then initiate a cascade of molecular events. And these molecular events can play extremely important functions in the cells and in our health, especially in many of the neurological diseases and neurological functions. And so there are a number of factors that can determine your NAD levels, and I have listed 4 of the most important ones. First of all, NAD is made from building blocks or precursors, and there are quite a few precursors that are on the market. The most famous ones are listed here. These are NMN, NR, or nicotinamide, and NAD can be de novo synthesized from amino acid tryptophan. So when we don't have enough building blocks, we certainly are not able to make sufficient NAD. And the second factor is there are several enzymes that are required to make the NAD molecule, and these enzymes can be deficient or suboptimal in many individuals, especially when we age. The third factor is what I call NAD degrading enzymes. And these are enzymes or proteins that require NAD as substrate for their function. And the most important ones are CD38, that has attracted a lot of attention recently. The PARP enzymes that are involved in DNA repair, and the sirtuins that play central roles in longevity. And especially CD38, because it is very abundant, it's present on the cell surface, and it's also present in the plasma. You can degrade the NAD quite rapidly and efficiently. And finally, secretion from cells into the circulation could be a very important contributing factor. You may have very high levels of NAD inside of your cells, but they may or may not be secreted into the plasma. So that can be a very important factor in determining whether someone has deficiencies or suboptimal levels of NAD in the plasma. So when it comes down to each person, you could be deficient in one or multiple of these pathways. So every person is different. That's why we need really a personalized approach in optimizing our NAD levels. There are multiple intervention strategies to optimize NAD levels, and the most popular ones are supplementation with precursors, and that's becoming quite popular recently. And you can also use supplements or nutraceuticals to increase the enzymes that can make NAD. And there are companies that are making and selling those supplements. And the third way to optimize your NAD is to inhibit the NAD degrading enzymes, such as the CD38 inhibitors, and like apigenin or 𝐶𝐶. And they can be effective, especially for circulating NAD. Now, NAD IV infusion it has been very popular for many years, and it's becoming increasingly popular. And whether what the IV infusion can or cannot do is a very important topic. And I do have a lot of data, but I'm not going to have time to go into those details today. I think you do need to know whether the IV infusion that you consider is serving the purpose for what you want to achieve. So I'll leave it at that for today. It's very important that we test in order to optimize NAD level. And so in order to optimize, we need to know the optimum range, and we need to define that. We also need to worry about potential toxicity. If you get your NAD levels to a very high level, that's not normally seen in the physiological conditions. And that's especially a concern when we talk about IV infusion, because you are putting a node of pure NAD into your blood. And these are the issues that have not been dealt with yet in our community. I think that those are very important. And testing before and after intervention is essential, and for you to know whether whatever you are trying to do is working or not, and whether you are at the optimum level. So NAD testing is very important. I don't think anyone would argue with me. But why NAD testing is so difficult? Why, we are the only company who is providing a NAD test that's available to consumers. There are primarily two reasons. The first one is NAD is not a very stable molecule. If the samples are not handled properly, and you can degrade very quickly. And secondly, most of the testing methodologies that are available use very expensive, high-end equipment that are usually not very reproducible, not very high throughput. So these are the two main reasons. At Ginfinity, we're able to overcome both of these issues through extensive research. And we have developed and we are offering two different types of NAD tests. One for circulating NAD in the plasma. And as you can see on the right, our assay is extremely reproducible, and you see a correlation plot between duplicated tests, and our R-square value is 0.97. Our average CV is 3%, and we do all tests in duplicate, so your CV is about 1.5% on average. So the test is works very, very well, and it's very stable. We also offer an intracellular NAD test, which is very easy. It's available worldwide, and the samples are collected on the filter paper that's used for newborn screening, and it's shown over here. So you basically do a finger prick, apply the blood on the filter paper. After a couple of hours, when it's dry, you apply a NAD fixing buffer to stabilize the NAD. You ship it back to us, and we will get it done, get it measured for you within a few days. So a lot of people ask me, what's the optimum range for NAD? I get this question a lot, so I want to address it here. So this is a very difficult question to answer. For the moment, I think the best thing that I can do is to take the distribution in a young age population. Here we are taking adolescents over 10 to 20 years old, and I'm taking the 25th percentile that we see in teenagers. So what it means is the 25th percentile means that 75% of the individuals have the levels. And here, the 25th percentile is at 34 micromole. So 75% of teenagers have an NAD level equal or greater than 34 micromole. So I take this value as the lower end of the optimum range. We can debate about this, but I think this is a very good starting point. We have not been able to define the upper limit of the optimum range. And my personal belief is you probably don't want to get it over 100 micromole, because over 100 micromole is not very often seen in any person in a natural way without supplementation or or NAD management. So I want to show you 2 examples that how we were able to increase the NAD level through precursor supplementation. Now this data I actually from my babysitter. She's 68 years old, and her starting NAD level was around 21 micromole or so. And then we tried a few different products on the market. It really didn't do a whole lot. And this one may increase a little bit, 10%, 15%. It doesn't do very much. Remember, we want the level to be above 34 micromole, above here. Then we started using another NAD booster that I cannot really disclose. And we got her level to over 100 micromole, 120 micromole. And that was great, it was interesting. But since then, I don't want her to get a level to above 100 micromole, and we decrease the dosage, and now her level is right around 50, 60 micromole. And so with the testing, we can really help you to choose the brand, the type of supplement, and the dosage to get your NAD level to a level that you want, that you choose. And we can really do that through testing. Here, this is my data from me personally. I have been testing myself from over a year ago. I started quite low. I'm 59 years old. I started around the 15 micromole. That's not shown here. And then I started managing it. With this particular supplement, I was able to gradually get my level to over 40 micromole. And then I stopped taking the supplement. You can see the level comes down gradually. Once I get it to below 34, I said stop it. I want to start taking supplement again. And I increased my dose. My level went up even more. I stopped it again to wash out so I can test a different supplement. And this is another supplement I tested. I got it to about 50 to 60 micromole. And so that's kind of where the sweet spot, I think, for me. And we are able to maintain this level pretty much, at the age. But you have to do it through testing, obviously. So, those are 2 cases that worked very well. So how about at the population level? So we tested hundreds of individuals before and after. As you can see, for most of the individuals, we were able to get their level to a very good range. Some are really high, but there are always a percentage of customers that cannot get their NAD to the optimum range the first time. So this is about 30% of the individuals. And they are taking different supplements, different approaches. And these are the individuals that need to change the types of supplement or the dosage, and or they actually need different approaches. There are a few individuals who have levels over 150. I have one person who got it to 250 micromole, and he works for a supplement company, and he wants to see how high he can get it to. 250 micromole is the world record, at least as far as I know. So I want to leave with this slide. I think if you really want to manage your NAD or manage your health, personalization is really the key. You really have to personalize. In order to personalize, you have to test, and then you have to take a personalized intervention. And after the intervention, you have to retest it again. So this testing, personalized intervention, retesting paradigm is so critical. I cannot emphasize more if we really want to have good efficacy, good outcome. There is no one-size-fits-all solution. we will have to experiment with ourselves. But now I think there are more and more tools available. There are more and more biomarkers available for you to really guide you and use a scientific approach to manage your health and manage your longevity. And I think I'll stop here, and thank you for listening.
Thank you so much, Dr. Ghee. I really appreciated the broader scope nature of your presentation today in comparison to the one you gave during our last biomarkers conference, though both were equally insightful. So thank you so much. Anybody who has questions for Dr. Ghee can pop them in the Q&A box below. We'll try and address them during the panel discussion. I'd like to welcome our next speaker to the floor, Jonathan Stein, Chief Scientific Officer and Medical Director at Testis Laboratories, who will be speaking on the topic of NGS and telomere testing and anti-aging diagnostics. Jonathan, a pleasure to have you with us. The floor is yours.
Okay, fantastic. Let me briefly talk. I'm going to discuss a couple of different anti-aging diagnostic tests that I developed for a couple of different companies. Start out with a quote. From Woody Allen. I don't want to achieve immortality through my work. I want to achieve it through not dying. I'm sure everybody here shares the same interest in living well and aging well. And one of the big things that shows up really quick is that not dying is sort of step one here. So let me briefly give you a bit of my background and sort of give you a sense of what I've been working on. I'm basically just a small town boy from Los Angeles. I got my PhD in molecular biology, essentially. UC Santa Cruz, which is the picture over here, a lovely place to visit if you ever get a chance. It's where the redwoods meet the Pacific. It's really nice. Got my PhD in postdoc at MD Anderson Cancer Center. From there, paternity forensic testing on the molecular side. Was the director of research for SpectraCell Laboratories for many years, and I'll present some data on the telomere testing we developed there. COVID hit like a truck, and I was all of a sudden consulting for 5 or 6 laboratories, and then from there became CSO and medical director at a number of different ones. I'm currently the Chief Science Officer and Medical Director at 𝐇𝐮𝐬 Laboratories, which is a next generation sequencing laboratory primarily, and I'm also the Medical Director at Gene by Gene. So I keep busy. And but my background is in developing assays and developing technologies, and I've always had a really keen interest in anti-aging. So let me jump right in. So just to focus the mind, again, if you want to age well, you need to not die first. And here are the things that Americans die from. This is the preliminary CDC data. They always have a lag time in December because they want to get it out early in January. But you can see from last year, your top 3: heart disease, cancer, and infectious diseases. Followed up a little further down by cerebrovascular disease, chronic lower respiratory disease. Here you're looking at pneumonias, influenzas, Alzheimer's, diabetes, and kidney disease. This will be important later when we talk about kind of biomarker development to identify metrics and measurements associated with risk and outcome with those diseases. Again, this is going to be a short and sweet talk, so I'll keep it quick. Please jump in and let me know if you have any questions. So there's lots and lots of different theories of aging, wear and tear, accumulative mutation, failure of the immune system, programmed senescence. All of that's there, and I'm a real yes and guy when it comes to this stuff. I'm pretty sure all of that's going on some level, none of them being particularly primary. And I'm sure I'm not the first person who's put up the blind men and the elephant analogy, where we're looking at different pieces of an overall problem and trying to map out what we're looking at with our assays, which are on a certain level very specific tools that only ask the specific questions they can ask, and then trying somehow to integrate that into a larger picture. with that in mind, and again, here's a slide that's probably come up more than once in this talk. There's lots of different models for aging at the cellular level. I posted a couple of links to some of them down here. There's a summary from the Cell paper. I think of this big picture as, you have these systems that are degrading over time. You have repair systems, which are themselves degrading over time. And if you can identify and map out those pathways, then you can start teasing out ways to modify those pathways. And interestingly enough, all of these different approaches, so here's some more frameworks for aging at the cellular level. Interestingly enough, all of these pathways describe the possibility for developing novel assays for them. Again, I'm an R&D guy, so I look at these and just see engineering opportunities. As we identify these systems, you're going to be able to, again, come up with assays for them. Some people do better, some people do worse, and then you have the opportunity to do back mapping or look at the outcome of interventions. You have all of these, once these tools are built and applied, you can do all sorts of really fun things with them. And I am definitely anti-aging for the fun of it. So, not to belabor this list, but it exists, and we can start tackling things from it. I developed this is from back in, 2008, 2009, something like that. I developed at SpectraCell a quantitative PCR-based telomere assay. And I'll again, very briefly go into some of the issues around it and some of the components of it and why it's interesting and important. So telomere lengths are associated with just about any kind of bad you can think of, and I'll post a more comprehensive list. All causes, morbidity, mortality, inflammation, cellular breakdown, the whole thing. And here's sort of what they are and what they're about. They're these repetitive DNA sequences that function essentially to cap and protect the ends of your chromosomes. The analogy everybody uses is shoelace covers, those little plastic tips at the ends of shoelaces called the aglets. So they function like that. And if cells lose that for whatever reason, the chromosomes will start to become wildly unstable. The thing that makes them interesting is the cellular clocks that over time, any reason that cells need to divide for growth, for repair, for replacement, because of oxidative damage leading to cell death, anything like that. Anytime a cell needs to divide, telomeres get a little bit shorter. And when they reach a sufficient level of shortness, the cells are given a signal to stop dividing. They're still alive, they just sit there incapable of additional division. It's called senescence. And this is thought to be a brake on uncontrollable cell division. So that's the idea behind it. Again, there's been something like 25,000 papers on this in PubMed. Shortened telomeres, shorter than average, are associated with all kinds of bad, just about anything you can imagine. Again, because any stressors on the body that result in large scale cell replacement are going to reduce your telomere lengths. So here's just a list of some of the ones that are involved. But honestly, you could think of just about anything that you can think of from a wellness perspective, and someone's done the paper and shown that that it will be associated with foreshortened telomere lengths, increased levels of stress, alcohol, like excessive alcohol and tobacco consumption. But you get some interesting, contrarian ideas about that as well that I'll talk about in a second. So again, so they work pretty well as cellular clocks. They function as growth regulators. I think I described a little bit about their mechanistic aspect. So I think of it, because there are enzymes that try to repair telomeres called telomerases. When you measure the average telomere length, what you're looking at is this sort of aggregate effect of an individual's wear and tear versus their repair, as well as the prospect of their future cellular replacement capabilities. So if someone's telomere length scores are really, really low, they are at risk for a whole host of other bad things, which again, I'll talk about in a second. But again, you've got repair mechanisms. When I originally set this up, and I think the business-minded among you will appreciate this, This was sort of before there was TA65, before people knew about astragalus, before people knew about interventions for telomere length. So the original ideas around it were somewhat fatalistic. Okay, well, here's this thing and it's slowly winding down and you can see where you are in your chart. And for about 2 years, the only people who ordered tests from SpectraCell were doctors and their spouses. So they were interested in their own telomere lengths, but didn't really know what to do with it or about it, or what the clinical utility of it was going to be. Well, the research kind of caught up. So a couple of things happened. Before I get to that, let me talk very briefly about one of the easiest models for this, right? So you've got these, one of the impact of telomere length on cardiovascular disease and stroke have been really well mapped out in big population studies. The impact of shortened telomere lengths on cardiac cells, impaired angiogenesis, the inability to grow new veins and arteries, myocardial muscle starts thinning out, and there's increased apoptosis, exactly what you'd expect, right? The cells can't divide anymore. So as your old cells are wearing out and they're waiting for the replacement cell to show up, they're not available because a bulk of cells have become senescent, and then eventually something blows out, right? Eventually something critical happens. So in a population study, I think there's been 2 large-scale ones, but in this one posted here, for every 1,000 base pairs a telomere was below average, someone's risk of cardiovascular disease and stroke went up about a little over 3-fold. So, so again, the mechanism is straightforward, and the outcome is sort of predictable based on this model of what telomeres are about. There's also an interesting aspect associated with cancer. They've done retrospective studies on cancer patients in large populations, and what they'll find is there's a subset of people for whom their telomere lengths are short, they're getting shorter, 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 and all of a sudden they'll bump right up. And what happens is, so as telomeres get really short, there's this braking mechanism to prevent uncontrolled proliferation, and for that to be bypassed, either cells will start pre-cancerous cells or cells on their way to becoming cancerous will either start overexpressing telomerase, the enzyme that repairs and lengthens telomeres, or the cells will become aneuploid, the number of chromosomes will change. They'll sort of rearrange their structure to bypass this telomere length break on cell growth, and then they'll start growing like crazy. Here is an example from breast cancer on the left, from breast cancer research, where as cells progress, you'll see shorter and shorter telomeres, and then all of a sudden they'll go up like crazy in terms of both telomerase activity and, though this slide doesn't show telomere length, we've certainly seen that to be the case in some cancers. In the right, they are looking at hematopathologies. And the interesting thing about this is that the telomere length assay is blood-based. Right, you pull peripheral blood from somebody's arm, you spin it down, you purify it out with all the nucleated cells, and you do a quantitative PCR assay on it to measure telomere length. And the interesting thing is we have identified in some cases pre-diagnosed people with leukemias and lymphomas because they had above human average telomere lengths, stuff that you might only see in a fetus. Or even higher than that. So in fetuses within our assay, you see, within newborns, somebody less than 1 year old, they're going to undergo massive expansion. And so they come out of the gate with telomere lengths in the 18,000 base pair longs or 18 kilobases. And if you see a 50 or 60-year-old person who has those scores, something is going really wrong. And so we've identified again, we've identified circulating metastatic prostate cancers, and while the test isn't diagnostic, and leukemias and lymphomas, while the test isn't diagnostic for that, it's certainly interesting that it all maps to the same results that we're seeing, which is to say, really low telomeres are terrible because, again, they are signifiers of lots of inflammation or lots of cell damage. Or the need for lots of cell repair, but higher than human average are also really bad because that means some underlying biological system is broken. and it's the one that acts as a brake on uncontrolled proliferation. So there's your sort of indication of cancer. It's interesting. So getting back to that notion of only doctors signing up, eventually the studies followed up and said, no, it can be improved. You can improve telomere lengths over time. And interestingly enough, it mostly falls under the same wellness rubric that you'd absolutely expect. You know, stop punching yourself in the lifestyle and things will get a little bit better. So the relative telomere length of, again, it's peripheral blood nuclear DNA, was about 5% longer among regular multivitamin users. So, you know, the assumption here is that people who are getting a boost in their antioxidants, you know, maybe they're taking less cellular damage from oxidative damage from reactive oxidative species. And so they have less cell death, so the less cell turnovers that are having, on average, longer telomeres. Moderate physical activity also showed, again, different assay, different measurement system, but they had about a 12% improvement on telomere length compared to both low and high exercise levels. Interestingly enough, one of the models by which more muscle gets built, myogenesis, is a cascade of oxidative damage from muscle cells, small muscle cells bursting during exercise. So people who engage in very intense exercise don't see any telomere length benefit from that. So this sort of makes sense intuitively because lots of professional athletes can die earlier of heart failure. And from the very small sample set, we have seen many of them sort of pay a price for very intense exercise in the absence of antioxidants. So, and that can all be ameliorated too, which is also good. Super interesting study back in 2008. They took a group of, this last bit down here, they took a group of prostate cancer survivors, put them on a fishitarian diet, had them doing yoga and meditation twice a week. So moderate exercise, stress reduction, huge improvement in diet because they were by and large standard American diet eaters. So not very healthy, went from not very healthy to very, very healthy. And within about 6 weeks, they saw about a 10% increase in telomerase and their telomere repair once they got that started. Quote down here, that which can be measured can be improved. So because we have this metric of telomere length and telomerase activity, we can look at corrective measures for it and see what's working and what doesn't work. During the course of time, I performed this testing. We certainly collaborated with different organizations that were looking to develop compounds and treatments that would improve telomere lengths. And some of them worked and some of them didn't. And until you can measure the thing, you don't know. You don't know. So another interesting one that probably very few of you have heard of is that SpectraCell performs, I should say, a test called Immunidex, which is a mitogen response assay. Mitogen response assays, they sort of came into prominence during HIV testing back in the 80s during the initial HIV epidemic, where they wanted to find out basically immunocompetence. The assay is really simple. You take peripheral blood from the patient, you spin it down, you purify out the nucleated cells, you put them on these 96 well tissue culture plates. So here's the spin down, you get this thing called a Buffy coat, it's nucleated cells. You spin them, you wash them, you plate them, and you grow them. Here, you stimulate the T lymphocytes to grow and divide, and T lymphocytes are basically the generals of the immune system. So you stimulate those cells to grow and divide, you let them grow for about a week, and you just see how well they grew. And that tells you how well a key component of your immune system can proliferate, can grow and divide when it's challenged to do so. And so that's a proxy assay for immunocompetence, right? And again, number 3 cause of death last year had a lot to do with your immune system. So that's the relevance to it. The interesting thing about this is that if you look at sort of this lower picture here of the Immunidex and this Immunidex versus age graph, you can see that on average, you see a slow and steady decline. but that there is, from here, a very large variance within testing populations. Some people have absolutely superb immunocompetence, and some people have terrible ones. And often that was linked to, because this is a functional nutrition testing company, it was limited, it was reduced typically by shortages of different key nutrients. So you can think of that as, you can think of your nutrients in this case as being building materials, right? Your immune system wants to build more cells, and if it needs zinc, or it needs selenium, or if it needs any trace micronutrient that's not available, then that growth is not going to happen as quickly or as effectively as if it was readily available. And so the interesting thing about this was, in general, we saw when patients were compliant and they they got their original score, they also got a list of nutrients they were deficient in, we tended to see a 15 or 20% improvement in their mitogen response, what we call the immunodex at SpectraCell. But their immunocompetence, its proxy measurement for immunocompetence, would improve by 15 or 20%, typically within 6 months of repleting deficient nutrients. So easy to address, really easy to address. And again, immune system is critically important, particularly as people age. And because there's lots of variance, you have good discriminatory power, you can identify how these can be improved, in some cases quite dramatically. So the last thing I wanted to kind of go into is my current position at 𝐇𝐮𝐬𝐧, and we're a next generation sequencing company primarily. We do a little bit of, what's that called, pathonc, a little bit of pathology, oncology as well. But again, primarily it's a molecular laboratory. We're looking at NGS. And here, I did a quick lookup before the presentation, unfortunately. So there have been genome-wide association studies looking at longevity, right? Twin studies, population studies. And the hope was that you'd find a bunch of, hopefully, some kind of hidden gems in the rough, right? Some very rare genes that have a what do you call it? A rare variant, and the rare variant either confers longevity or removes it, and that would identify pathways that could then be, targets for either biomarker development or for therapeutic intervention or lifestyle intervention, anything like that. But unfortunately, at least in this one particular study, most of heritability was employed by common genetic variants. So no silver bullets on this one at present, but at least in the paper, they're going to keep digging. So we'll see what comes out of that. We do NGS panels for cardiovascular and cerebrovascular disease, cancer, Alzheimer's disease, dementia, and diabetes. There's talk in the works for looking at pulmonary infectious disease panels. We do pharmacogenomics testing to reduce the likelihood of iatrogenic injuries or injuries caused by treatment. So, making sure that people are getting the right amount and the right dosage of medication, not too much, not too little, finding their individual Goldilocks zone for that. And there's certainly research in the works for tumor characterization panels and sort of molecular basis of cancer testing. So all of those technologies, kind of come into place when we're looking at reducing these sorts of kind of frontline causes of death, right? Identifying people who are at high risk of cardiovascular disease and stroke, and developing interventions for them. At least identifying people who are at higher risk of cancer and figuring out how to address them. Beefing up the immune system. Looking at, again, stroke risk, again, tumors associated with stroke risk, looking at beefing up the immune system for influenza and pneumonia, looking at markers for Alzheimer's disease and diabetes, so that we can identify people at risk and look at intervention methods and maybe pharmacogenomic pathways or targets of intervention there. So that's kind of what I've been up to lately. Wanted to just conclude a quick Greek myth about Eos and Tithonus. as a kind of way of getting to a notion about health span. So this is an old Greek myth. Eos was the dawn. She was beloved of Tithonus. The gods granted Tithonus immortality, but he still aged. He was just going to live forever, but he continued to normally age, and eventually he became a cicada, which greets the dawn, which I don't think is in anybody's bucket list. Far more likely that we would all want to be this guy, be old, but to be in vigorous health, and to be living well and have as long a health span as we can. So that's sort of the goal set there. And I'll thank you. And if you guys have any additional questions, here's my work email. Thank you for your time.
Thank you so much, Jonathan. The pleasure was all ours. I'd now like to welcome to the floor our final speaker for today prior to the panel discussion, our very own Kate Batts, Director of Femtech Analytics and a fellow partner of Longevity Capital. Kate, always great to have you with us. The floor is yours.
Thank you very much for the introduction, Franco. Well, first of all, it's a pleasure to be here, and I thank all of our speakers for giving us excellent presentations. I have been a proud member of Deep Knowledge Group for almost 3 years now, and I'm very excited and privileged to be a director of our new analytical subsidiary, Femtech Analytics. We've started in June of this year, and we're very active in the field. Many of you know that Deep Knowledge Group always deeply cared about longevity or health span, and Femtech Analytics, focuses specifically on female health. I would like to, provide a brief overview of our recent open access analytical report, which is part of the broader longevity biomarkers report, which we were fortunate to collaborate with together with Longevity International and Aging Analytics Agency. So I'll start with the Femtech industry review, briefly touch upon biomarkers classification, focus on biomarkers of longevity, and give some key conclusions, as well as a brief update as to what are we up to. So what is Femtech? Femtech generally refers to software, diagnostics, products, and services that use technology to support women's health. The projected growth rate is actually quite remarkable. So it is estimated to have been 21.7 billion in 2020, and it is projected to reach 60.1 billion in 2027. One of the key reasons behind that is actually a shift in socio-cultural paradigm of how female health is viewed. Traditionally, the most important sector was involved with female reproductive health, but we are glad to see that there is an expansion of the umbrella of Femtech that now involves, mental health, sexual well-being, and a number of other sectors. As part of our report, we also prepared an open access analytical platform that everyone is welcome to use if you go to femtech.health/report, because we offered our classification of Femtech, and we profiled over 1,500 companies, 1,000 of investors, and a number of R&D centers. In terms of our Femtech biomarkers classification, first of all, these are biomarkers that are specific to women only. As I mentioned, reproductive health does play an important, role here, and is the leading segment. So we definitely have pregnancy biomarkers such as pregnancy trackers, prenatal testing, neonatal testing. Also biomarkers of female cancers such as breast, ovarian, cervical, endometrial. We also have biomarkers of female reproductive health, such as vaginal microflora, endometriosis, infectious disease biomarkers, and biomarkers of pelvic floor disorders. And there's also, of course, fertility biomarkers such as ovulation trackers, menopause biomarkers, hormonal biomarkers, and ovarian reserve biomarkers. And finally, there are biomarkers of general women's health, such as complex health assessment, biomarkers of cardiovascular diseases, and skin biomarkers. This diagram actually depicts the key companies, investors, and R&D sectors working on the respective subsegments here. I've mentioned earlier that longevity has been our long-term focus and remains to be such. Aging affects all of us, and women live, while they live longer, they do not necessarily live healthier. And the longevity biomarkers are especially important here, and we separate them in three key groups. One is the general women's health biomarkers, including skin, cardiovascular health, as well as complex health assessment. We do have reproductive health biomarkers, of course, including infectious diseases, endometriosis, reproductive aging, and age-related diseases, as well as cancer biomarkers that I mentioned earlier. Speaking of cancer, approximately 13% of women suffer from it, but thankfully, a lot of it can be prevented. In terms of biomarkers of cancer, breast cancer leads in terms of the focus, followed by ovarian cancer, cervical cancer, and endometrial cancer. Pregnancy biomarkers are dominated by pregnancy trackers, prenatal tests, and neonatal tests. Fertility biomarkers, essentially assess a woman's capability of getting pregnant and giving birth. Here, we have fertility hormones leading the way, followed by ovulation trackers, ovarian reserve trackers, and menopause detectors. Reproductive health-wise, we have the important sector here is infectious diseases, especially HPV. And many companies also develop vaginal microflora biomarkers as well, followed by endometriosis biomarkers, because here early prevention, detection, and is extremely important. Also, I mentioned pelvic floor disorders biomarkers as well. And then going to the biomarkers of general women's health and longevity, the important one here is skin health, followed by cardiovascular health, and a complex health assessment. In terms of market overview as of this hour, we would see that in terms of specialization, more than 50% of companies in the space are involved in the fields that concern reproductive health, fertility, and pregnancy biomarkers. But I mentioned earlier that we now have also other sub-segments in the space. And then the other 30% of the companies are focusing on various female cancers. If we take a regional overview, United States is a clear leader, followed by Europe, Israel, and India. This chart actually represents the companies that are leaders in terms of funding, and I encourage everyone to check out our report, because I don't want to delve into too much details as we are pressed for time. R&D centers, United States leads as well, followed by UK, followed by Europe, and Canada. Investors-wise, again, North America is in the lead, followed by Europe. There is a welcome trend of a growing number of femtech companies, and they are focusing on the segments that I've mentioned earlier, and there's a steady growth. In terms of conclusions, femtech is definitely an emerging field. It's growing fast. It has a high compound annual growth rate. It combines both solutions and startups that focus on women's health. Fertility and pregnancy issues remain to be, the leading sectors, but there is an increased focus on other aspects of female health as well. And femtech biomarkers, in our view, refer to four main areas of women's life. It's general health, reproductive health, fertility, pregnancy, and women's cancers. And the most significant numbers of femtech biomarkers companies, as well as R&D centers, are in North America. So a brief overview of what are we up to. Those of you who were at our conference yesterday probably had a chance to see Dmitry's presentation on his book on biomarkers of human longevity, which I highly encourage everyone to read. And we're going to get the PDF once we conclude our conference. There's an upcoming book that our team is preparing that will focus specifically on women longevity, and it's coming out later this year. And our analytical subsidiaries, traditionally, starting from Aging Analytics Agency, have been producing a number of open access analytical reports, which are accompanied by IT platforms. Femtech Analytics is no exception to this rule. And since its inception in June of this year, we actually produced a number of open access analytical case studies, such as Femtech Industry Overview, Investment Digest, and a number of other projects, which I encourage everyone to take a look at www.femtech.health. Further, we're also going to invest in the space. We are in the process of setting up a special purpose vehicle, which will be investing in Femtech. And it's good news that in 2020, a total of 520 million of VC capital was invested into Femtech. And we hope that this trend will continue. And it's grown tenfold since 2012. It's been a pleasure to give this presentation. I thank everyone who is attending our event. We can be easily reached at femtech.health. Feel free to shoot me an email. I'm also pretty active on LinkedIn. Thank you.
Thank you so much, Kate. I'd now like to welcome all of our remaining speakers to join us, switch on their videos, and participate in a panel discussion, which will be led by Kate. So Kate, the floor is yours.
Thank you. Well, first of all, I would like to thank our speakers again. I know it's pretty late in some of your locations. This is a live session, so we do have, a live audience. So I encourage everyone to drop questions in the Q&A session, and I will ask Franco to intervene if there will be some interesting questions there. And I think that one of the important takeaways of today's panel discussion is that there's such a wide variety of biomarkers and its practical applications. And it is wonderful to see what a positive effect it has on aging research and how it advances our longevity industry as a whole. I think also that with the rise with the increased focus on precision health, longevity biomarkers will become increasingly important, both for R&D, success of the companies, as well as the overall health of the population. So with that, I will ask a few questions, and I encourage our speakers to raise questions as well if something appears of particular interest. So I will start with that. We know that today there is a wide range of existing biomarkers and their practical applications. What, in your view, are the new emergent trends of new biomarkers, and what role will AI play in their detection and validation?
Well, I would welcome both Anastasia and Ryan to field this, because they both gave very exciting presentations about rather new multimodal biomarkers. In the case of Anastasia, it was image-based. In the case of Ryan, it was voice-based. On the topic of AI, I think that most of us would agree here that AI is going to play a not just complementary but necessary role in all of this. There's the volume of biomarker data and the new types of biomarkers that are being validated. This is just going to continue growing. It's already fairly mountainous, fairly unapproachable by humans. So AI is necessary, and that trend's only going to continue, both from a raw volume of data perspective and from a multimodal data perspective, trying to integrate and synthesize all of these multimodal biomarkers. But with that being said, Anastasia, Ryan, do you have any comments?
Yeah, if you don't mind, let me probably start. I basically think that the very idea for us having multiple biomarkers is being able to get actionable advice and assessment of our health condition or our longevity status as soon as possible. So I personally see that in the recent decade, we see that there's happening a shift from, I would call them more traditional biomarkers, like coming from epigenetic logs, methylation, using other types of omics data towards exploring other data that should address, address criteria that Franco you have highlighted. So first, it's availability of the data and being able to standardize it, as I think I really tried to highlight standardization in my presentation. So these types of data are massively enabled by multiple trackers and different services to track, for example, weather condition. And I personally think that being able to use different devices, different wearables is becoming a big trend. So you even can see within the cosmetics area. So L'Oreal released a UV tracker. And talking about, where it logically goes, I think that since we're getting more and more biomarkers, it does make sense to combine them into functional groups. For example, your movement biomarkers, like heard the presentation from Gero talking about activity biomarkers, or it could be your skin biomarkers, or it could be female health-related biomarkers, or maybe we will address not only the physical biomarkers, but biomarkers of our mental health. So how, what are our decisions? So what longevity horizon do we envision? So we are thinking, and I think that's basically a way to go. So getting a look at more available data for wearables and connected devices, and at the same time exploring other alternatives to traditional omics biomarkers. And I think today we had amazing presentations highlighting a lot of new alternative and a lot of promising biomarkers.
Thank you.
And I'd be happy to follow up on that, at least briefly, just to say that I think that in our investigation, we would absolutely agree that the multi-omic biomarkers are the way to go. I think that looking at any one particular platform always leaves, I would say, some additional questions. And I think that looking at a more comprehensive data set altogether is extremely helpful. And particularly, in the epigenetic space, there recently was some publications on ways to measure methylation in the brain due to methylation-specific diets and MRIs. I think that's particularly exciting as well.
Great, thank you. Well, speaking of continuing the topic of AI, what do you think will be its key role? Will it be finding new biomarkers or assessing the existing ones?
Yeah, to sort of continue for that, I'd love to jump in there as well. I think that definitely, right now, even from a methylation specific standpoint, at the largest scale platforms for sort of array-based technology, we're only looking at 850,000 approximate CpGs out of the 29 million CpGs per cell, and most of the investigation has been carried around only in blood. And so by far, I would say that our investigations, and even the methylation clocks that exist today, are only a fraction of what they could be in the future. And so I think that definitely new algorithms, finding new biomarkers from advanced data sets and more robust data sets are really hold the hope to some of the best clinical biomarkers. I think that, every day almost there's a new algorithm that comes out that has some particular advantage over the others. And I think that type of innovation will not stop anytime soon.
Great, thank you. Does anyone else have any other comments here?
Just briefly, that it's probably going to be a bit of both, of course. The volume of existing biomarker data, we've just scraped the surface, the tip of the iceberg, as Ryan mentioned. Besides that, we're seeing an explosion of new categories and types of biomarkers being validated as being useful. AI is going to be critical for both of those. Yeah.
And maybe just as last comment, probably the third thing where AI can help will be to also identify the interrelations and the causalities and functional relations between those biomarkers, and hopefully also create, as we mentioned the first question, a subset or a cluster or some sort of multifunctional biomarker cluster. And hopefully also AI will help us to somehow at least pre-test the validity and and efficacy also and applicability in the clinic. So the in-clinical trials that are already existing, hope this will also evolve and become more of a standard, and become more of a tool that will allow us to, pre-validate the markers.
Could not agree more, Evelyn. AI gets a lot of spotlight for accelerating drug discovery per se, but equally exciting things are happening on the level of biomarkers as well. So yeah, great point.
Well, and again, a question to the panel: what type of biomarker do you think is likely to be the most actionable and the most successful one, or the group of biomarkers rather?
I think following up on what Ryan just said, I think it's all about the interconnectability of different biomarkers. I think there will not be the one digital biomarker. There will be a group of disease and condition specific biomarkers. And just to mention another point here in this discussion, I think it's also about the regulators to give us a very clear framework on how to medically validate clinical grade biomarkers. I think this is one of the most important things in the future, especially when it comes to digital biomarkers, having a really clear framework on how to validate and being clinically accepted in the end. I think this is quite important here.
There is a pressing need for highly actionable biomarkers, very often biomarkers which are good enough, but which are non-invasive, cost-effective, highly scalable by certain stakeholders, whether it's a company or whether it's a national government trying to implement longevity policies, etc., like the UK is doing with their goal to add 5 years of healthy life expectancy to their population by the year 2035, etc. Actionability is often a better trade-off than accuracy. That being said, I'm a big fan of blood-based biomarkers, especially when coupled with AI and deep learning, though I'm biased because I've published a few papers on blood-based biomarkers. But that being said, we're also seeing an explosion, as viewers are probably aware, of digital biomarkers and biomarkers which are highly obtainable through consumer devices like voice, like image, etc. It's really great to see these things stepping up in order to meet that pressing need. And indeed, the new analytical report from Aging Analytics Agency produced for Longevity International, which we launched yesterday. 200 of the 300 companies profiled there have at least one market-ready product, whether it's consumer-based or in the clinic. So
let me jump in. So all the biomarkers that we offer at Ginfinity Precision Medicine are actionable now. And we have 13 biomarkers that we offer to consumers. And we probably have a dozen or so more biomarkers that we offer to researchers, but not to consumers, because I don't think they are quite actionable yet. So let's discuss what biomarkers are actually actionable. So you need at least two very basic fundamental elements for a biomarker to be actionable. The first one is on the nature of the biomarker, the validity of the biomarker. Are the biomarkers clearly validated by sound scientific studies and large scale, not just on a few patients? That can be often the case. So very well clinically or scientifically validated biomarkers. That's the requirement number one. Requirement number two, you must have solutions to act upon this information. And you can have very well validated biomarkers, but if there's nothing you can do, that's not going to be actionable. So once you have these two essential elements in place, a biomarker becomes actionable. And there are many actionable biomarkers, fortunately, but many of the newer biomarkers that are being developed will probably have to work on both fronts for them to become more actionable. A lot of progress are being made, but we're not quite there for some of the biomarkers yet.
Great, thank you. Well, we mentioned today the silver tsunami or the age wave. There are different colloquial terms for that. So, and we know that the chronological age of global population is increasing due to advancements of medicine. So, what about biological age? Is there, any opinion on that?
So, okay, if I may just, try to rephrase your question. So, is your thinking around how is our population becoming biologically younger, or did I get the question wrong?
Well, it's a question. So we know what is the demographically chronological age of the population, but are we becoming? What is your opinion about the biological age of?
Sure. Okay. Well, let me probably try to answer this question, but I think I will be happy if colleagues will join me, because obviously I'm more focused, on skin aging. But overall, I think it was well depicted today on our conference and this panel that primarily a lot of stress and adverse effects of our environment, they have a very big impact on our longevity status, and we see that our biomarkers do decline. So this is why we now know that we need a good meditation, a good exercise, a good diet. I overall think that we now see that in many of well-developed countries, we see that life expectancy is rising, and since we're consciousness of patients is raising, awareness with our techniques that allow them to extend longevity. I would say that in this sense, chronological age of the population, so the population is aging, it's a well-known fact, right? So we ideally want to somehow mitigate the aging population with chronologically aging population, with the fact that we at least can stay at the same time biologically. It's very difficult, to reverse. At the first place, we need to be able, to stop aging. So I think the overall trend will be that we will live longer life, we will live more quality life. So in that sense, the population biological age ideally should decrease over time. However, we had really striking events the last 2 years, which I think dramatically decreased life expectancy, dramatically decreased the quality of life. A lot of planned operations and planned care was postponed and delayed for the time, but we even cannot foresee for how much time it will take longer. So I would say if we are able to survive this crisis, then probably we will, in the time frame of maybe 5-10 years, see that our population will gradually become biologically younger. So that's my perspective, and I'm happy if colleagues will join me here.
I would just like to say that it's, when we're talking about population level health, it's a highly multivariable issue, and it's very hard to give a clear answer, Kate. But I would generally agree with Anastasia. We're seeing a lot of longevity therapies, technologies, etc., behavioral interventions being validated to an increasing extent, entering market readiness via either the clinic or consumer product, etc. And this, when coupled with the rising interest of population health and actual longevity initiatives by governments, which is a thing. We're entering the age of longevity politics, which is not the topic of today, but factors in here. Yeah, I think that we'll see improvements on a population level, at least at the aggregate. Evelyn, I saw that you unmuted yourself at the beginning of this question. Did you want to add something?
Yes, I think it's a great question. So I believe rather than seeing a reversal for the upcoming realistically years, we will see some other shift, hopefully, which is implementing biological age first as the definition of age in aging, right? So we have now achieved, thanks to also Franco, one of your great achievements, right, with other colleagues, was to actually give us clinicians now the chance to categorize within ICD-11 old age and aging, at least as as a disease or as a symptom, and then also, as an addition, causality. So that's wonderful. And now I think the debate is going towards how do we define aging in measurable terms, and thanks to biomarkers, now we know that we can measure biological age. So I think the shift would be on a population public health level, hopefully towards classifying diseases, age-related diseases, as related to biological age, and also then allowing biological age to be some sort of a predictor, for diseases. So again, for example, in oncology, but also in other like in neurodegeneration, or even in infectious diseases. So we saw again with COVID, I couldn't agree more that age is related to it. So measuring biological age, and then I also agree with Anastasia that hopefully we'll see more and more of a reversal and slowing down of the biological age. That will be a big task of longevity medicine to achieve that at some point of time, also on the global and population level. Ultimately, of course, by the statistic, we will see a growing, population also with a growing biological age, because we cannot expect, I guess, living forever. So keeping the biological age always at a low level, but it will always be probably something that that the trend will be the same. We'll see a tsunami of people who are coming with a silver biological age, because we are all aging. But the shifts that should happen on a global level, global health level, would be rather to redefine age and aging towards biological age as a surrogate.
Yeah, very interesting perspective, and I couldn't agree more. And thank you, by the way, for your kind comment about the ICD-11 work that we did, adding the new extension code for age-related diseases via ICD-11 in 2018. Appreciate it.
Well, Franco, I know we're slightly running over. It's been a great pleasure to meet the influencers in the sphere, and we would like to continue holding these events. Biomarkers are actionable, are very important in R&D and how it translates to success of the companies in the space, as well as overall human health. So I'm very happy that I was part of this event. I definitely learned a lot, and I hope we will meet again in not so distant future. And thank you everyone for attending, and for the audience for being here as well.
And if you'll allow me, I have a few closing remarks. So we are about an hour, almost an hour over time, but it was a very exciting 4 hours. I feel a little bit younger and a little more awake, believe it or not, at the end of this. So yes, tremendous thanks to all of our speakers and attendees for joining us today. Everyone who's in attendance today can expect a follow-up email with the full and individual video recordings of both sessions yesterday and today. Once again, a big thank you to all of our speakers. It was a pleasure hosting and moderating you. And I'm glad that we were able to extend things a bit to actually have this panel discussion. And thank you so much for bearing with us time-wise in that regard. I'd also like to ask everybody to keep an eye out at longevity.international, both for the individual video recordings. We're going to be setting up a landing page to hold all of that, but also for new updates, new reports, new event announcements, etc. This is not the end of biomarkers of human longevity for us from a broad activity perspective or from a conference perspective. We do have some things planned, which we'll be rolling out and announcing in the coming weeks, months, etc., both in an online and physical format. So please be subscribed if you like to longevity.international to keep an eye out for that, and stay tuned. And for everybody viewing today, in case they missed it, once again, I'd just like to remind you that we launched 2 important publications yesterday. The first being Aging Analytics Agency's newest and third Longevity Biomarkers Landscape Overview, Q4 2021, in an open access format for Longevity International, in order to try and maximize the viewership and the fruits of our analytics for various industry stakeholders. And finally, yesterday, we also launched the newest book from our general partner, Dmitry Kaminskiy, Biomarkers of Human Longevity, Data Science for Accelerating Aging Research and R&D, the critical catalyst for practical human longevity, tangible investment decision-making, and de-risking. I was told yesterday that all of today's participants and viewers will be sent a free interactive full-color PDF of that book as a follow-up via email in the coming days. So look out for that. I highly recommend that you read it. Thank you one and all. Once again, it's been a pleasure, and I hope to see you again soon.